Microbiology, An Introductory Guide to Microorganisms. By Ray Arders. Chapter 1, The Invisible Universe. Microbiology reveals a vast universe of life that exists largely beyond the reach of our unaided senses, encompassing billions of microorganisms that inhabit every conceivable environment on Earth and profoundly influence all aspects of life as we know it. This hidden world includes bacteria, archaea, viruses, fungi, and protists that collectively represent the most abundant and diverse forms of life on our planet, having evolved over billions of years to occupy virtually every ecological niche from the deepest ocean trenches to the highest atmospheric layers. The discovery of microorganisms fundamentally transformed our understanding of life itself, beginning with Antony van Leeuwenhoek's first observations of animalcules through his primitive microscopes in the 17th century. These initial glimpses into the microscopic world revealed a complexity and abundance of life that challenged existing concepts about the nature and distribution of living organisms, ultimately leading to the recognition that microbes represent the most ancient and fundamental forms of life on Earth. Cellular diversity among microorganisms encompasses the full spectrum of cellular organization, from the relatively simple prokaryotic cells of bacteria and archaea that lack membrane-bound organelles, to the complex eukaryotic cells of protists and fungi that contain specialized subcellular compartments. This diversity reflects billions of years of evolutionary experimentation that has produced an extraordinary array of cellular architectures and biochemical capabilities that far exceed those found in multicellular organisms. Prokaryotic cell structure demonstrates remarkable efficiency in organizing essential cellular functions within a relatively simple organizational framework, with the bacterial cell envelope serving multiple functions including structural support, selective permeability, and environmental sensing. The peptidoglycan cell wall provides mechanical strength while allowing for controlled expansion during growth, while the cytoplasmic membrane regulates the passage of molecules and maintains the electrochemical gradients essential for energy generation. Archaeal cells, while superficially similar to bacteria in their prokaryotic organization, exhibit fundamental differences in cellular components and biochemistry that reflect their distinct evolutionary history and adaptation to extreme environments. The unique lipid compositions of archaeal membranes, distinctive ribosomal structures, and novel metabolic pathways demonstrate that archaea represent a separate domain of life with characteristics that are neither typically bacterial nor eukaryotic. Eukaryotic microorganisms showcase the evolutionary innovations that led to complex cellular organization through the development of membrane-bound organelles that compartmentalize cellular functions. The endosymbiotic origin of mitochondria and chloroplasts represents one of the most significant evolutionary transitions in the history of life, enabling eukaryotic cells to harness aerobic respiration and photosynthesis while laying the foundation for multicellular complexity. Viral particles occupy a unique position in the biological world, existing at the boundary between living and non-living matter, as obligate intracellular parasites that require host cells to reproduce. The diversity of viral structures, ranging from simple helical and icosahedral forms to complex architectures with multiple protein layers and appendages, reflects the varied strategies that viruses have evolved to infect different types of host cells and exploit cellular machinery for their replication. Microbial size and scale present both opportunities and constraints that shape the fundamental biology of microorganisms, with high surface area to volume ratios, enabling rapid exchange of materials with the environment while limiting the complexity of internal organization. These physical constraints have led to evolutionary innovations, including specialized transport systems, efficient metabolic pathways, and sophisticated regulatory mechanisms that allow microorganisms to thrive despite their microscopic dimensions. Microscopy techniques have evolved dramatically since Leeuwenhoek's simple lenses, with modern instruments enabling visualization of cellular structures at molecular resolution and revealing the dynamic nature of microbial life. Light microscopy remains essential for basic microbial observation, while electron microscopy provides detailed views of cellular ultrastructure, and advanced techniques including fluorescence microscopy and atomic force microscopy enable real-time observation of cellular processes and molecular interactions. Cultivation methods for microorganisms have expanded from simple broths and solid media to sophisticated systems that can reproduce specific environmental conditions and support the growth of previously unculturable organisms. The development of selective and differential media has enabled the isolation and identification of specific microbial species from complex communities, while enrichment techniques allow researchers to favor the growth of organisms with particular metabolic capabilities. Molecular techniques have revolutionized microbiology by enabling the study of microorganisms without requiring their cultivation, revealing that the vast majority of microbial species have never been grown in laboratory conditions. DNA sequencing technologies have uncovered enormous microbial diversity in environmental samples, while techniques such as metagenomics enable the study of entire microbial communities and their collective metabolic capabilities. Classification of microorganisms reflects our evolving understanding of microbial diversity and evolutionary relationships, with traditional morphological and physiological criteria being supplemented by molecular analyses that reveal deeper evolutionary connections. The three-domain system recognizing bacteria, archaea, and eukaryotes represents our current best understanding of the fundamental divisions of life, though ongoing discoveries continue to refine and expand this framework. Microbial genetics demonstrates the remarkable plasticity of microbial genomes, with horizontal gene transfer enabling rapid acquisition of new capabilities and adaptation to changing environmental conditions. The compact organization of microbial genomes, with high gene density and minimal non-coding DNA, reflects the selective pressures for efficient resource utilization, while maintaining the genetic flexibility necessary for survival in diverse and changing environments. Biofilms represent a fundamental mode of microbial existence where cells aggregate within self-produced polymer matrices that provide protection from environmental stresses and enable complex community interactions. These structured communities exhibit emergent properties, including enhanced resistance to antimicrobial agents, sophisticated communication systems, and division of labor among different cell types within the biofilm structure. Quorum sensing enables microorganisms to coordinate their behavior based on population density through the production and detection of chemical signaling molecules. This communication system allows microbial communities to synchronize activities such as biofilm formation, virulence factor production, and metabolic processes, demonstrating that even single-celled organisms can exhibit collective behaviors typically associated with multicellular organisms. Chapter 2: Microbial Metabolism and Physiology. Microbial metabolism encompasses an extraordinary diversity of biochemical pathways that far exceeds the metabolic capabilities of any other group of organisms, enabling microorganisms to exploit virtually every available energy source and carbon substrate, while thriving in environments that would be lethal to most other forms of life. This metabolic versatility reflects billions of years of evolutionary innovation that has produced novel enzymatic systems and metabolic strategies unique to the microbial world. Energy generation in microorganisms involves diverse mechanisms for capturing and converting energy from chemical, light, and even electrical sources into the universal cellular currency of ATP. Chemotrophs derive energy from chemical reactions involving organic or inorganic compounds. Phototrophs harness light energy through various photosynthetic systems, and some microorganisms can even extract energy from electrical currents or magnetic fields, demonstrating the remarkable adaptability of microbial energy metabolism. Aerobic respiration in microorganisms follows the same basic principles as in higher organisms, but exhibits greater flexibility in the types of substrates that can be oxidized and the terminal electron acceptors that can be utilized. Many microorganisms can switch between aerobic and anaerobic metabolism, depending on oxygen availability, while others have evolved specialized respiratory chains that can utilize alternative electron acceptors such as nitrate, sulfate, or metal ions. Anaerobic respiration enables microorganisms to generate energy in oxygen-free environments through electron transport chains that utilize alternative terminal electron acceptors, creating opportunities for life in environments where aerobic organisms cannot survive. These processes are crucial for global biogeochemical cycling and include denitrification, sulfate reduction, and metal reduction that transform inorganic compounds while generating energy for cellular processes. Fermentation pathways allow microorganisms to generate energy through substrate-level phosphorylation without requiring external electron acceptors, enabling survival in completely anoxic environments while producing diverse end products that have important ecological and industrial applications. The variety of fermentation pathways found in different microorganisms reflects their adaptation to specific substrates and environmental conditions while producing characteristic waste products. Photosynthesis in microorganisms includes both oxygenic photosynthesis, similar to that found in plants, and anoxygenic photosynthesis that does not produce oxygen as a byproduct. Cyanobacteria perform oxygenic photosynthesis using two photosystems that enable them to use water as an electron donor, while purple and green bacteria use simpler photosynthetic systems that require reduced compounds such as hydrogen sulfide or organic molecules as electron donors. Chemosynthesis represents a uniquely microbial form of primary production, where energy for carbon fixation is derived from chemical reactions rather than light, enabling the existence of ecosystems in environments completely isolated from solar energy. These processes include the oxidation of ammonia, sulfur compounds, methane, and hydrogen that provide energy for carbon dioxide fixation while supporting entire food webs in environments such as deep-sea hydrothermal vents. Carbon metabolism in microorganisms demonstrates remarkable flexibility in the sources of carbon that can be assimilated and the pathways used for carbon fixation and utilization. Autotrophic microorganisms can fix carbon dioxide through various pathways, including the Calvin cycle and alternative carbon fixation mechanisms, while heterotrophic microorganisms can utilize an enormous diversity of organic compounds ranging from simple sugars to complex polymers and even synthetic compounds. Nitrogen metabolism involves specialized microbial processes that are essential for global nitrogen cycling, including nitrogen fixation that converts atmospheric nitrogen gas into ammonia, nitrification that oxidizes ammonia to nitrite and nitrate, and denitrification that reduces nitrate back to nitrogen gas. These processes are performed exclusively by microorganisms and are crucial for maintaining the availability of nitrogen in forms that can be utilized by other organisms. Extremophile physiology reveals the remarkable adaptations that enable microorganisms to survive and thrive in conditions that would destroy most other forms of life, including extreme temperatures, pH values, salt concentrations, pressure, and radiation levels. These adaptations involve specialized proteins, membrane compositions, and metabolic pathways that maintain cellular function under conditions that approach the theoretical limits for life. Thermophile adaptations include heat-stable proteins with increased numbers of ionic bonds and hydrogen bonds, modified membrane lipids that remain stable at high temperatures, and specialized chaperone proteins that assist in proper protein folding under extreme heat. Some hyperthermophiles can grow at temperatures exceeding 100 degrees Celsius, demonstrating the remarkable plasticity of biological systems. Acidophile mechanisms for surviving extremely low pH environments include specialized transport systems that pump excess protons out of the cell, buffer systems that maintain internal pH homeostasis, and acid-resistant proteins and cellular components that can function in highly acidic conditions. These organisms play important roles in metal extraction and acid mine drainage, while demonstrating the limits of biological pH tolerance. Halophile strategies for surviving high salt concentrations include the accumulation of compatible solutes that balance osmotic pressure, specialized proteins that require high salt concentrations for proper folding and function, and modified cellular membranes that remain stable in hypersaline conditions. Some extreme halophils require salt concentrations approaching saturation for optimal growth. Psychrophile adaptations to cold environments include antifreeze proteins that prevent ice crystal formation, modified enzymes that remain active at low temperatures, and specialized membrane compositions that maintain fluidity in cold conditions. These organisms are crucial for biogeochemical cycling in polar environments and may provide insights into the possibility of life in cold extraterrestrial environments. Radiation resistance in some microorganisms involves sophisticated DNA repair systems, protective pigments, and antioxidant mechanisms that enable survival of radiation doses thousands of times higher than would be lethal to most organisms. These capabilities have potential applications in bioremediation of radioactive environments and space exploration. Microbial stress responses demonstrate the sophisticated regulatory systems that enable rapid adaptation to changing environmental conditions through the coordinated expression of genes involved in stress tolerance, DNA repair, and metabolic adjustment. These responses can involve global regulatory networks that reprogram cellular metabolism and gene expression in response to specific stress conditions. Regulatory mechanisms in microorganisms include transcriptional control systems, post-translational modifications, and small RNA molecules that enable rapid and precise control of cellular processes in response to environmental conditions. The efficiency and speed of microbial regulatory systems enable rapid adaptation to changing conditions while minimizing the energetic costs of unnecessary protein production. Metabolic engineering applications exploit the metabolic versatility of microorganisms to produce useful compounds including pharmaceuticals, biofuels, and industrial chemicals through the modification of existing metabolic pathways or the introduction of novel pathways into microbial hosts. These approaches represent important sustainable alternatives to traditional chemical synthesis methods. Chapter 3: Microbial Diversity and Ecology. The diversity of microbial life exceeds that of all other forms of life combined, encompassing millions of species that occupy every conceivable habitat on Earth, while exhibiting an extraordinary range of morphologies, metabolic capabilities, and ecological strategies. This diversity reflects the ancient origins of microbial life and the billions of years of evolutionary innovation that have produced organisms adapted to virtually every environmental condition found on our planet. Bacterial diversity encompasses an enormous range of morphologies, metabolic capabilities, and ecological roles that demonstrate the evolutionary success of prokaryotic life. Major bacterial phyla include the Proteobacteria, which contains many familiar organisms including Escherichia coli and various pathogenic species, the Firmicutes, which includes endospore-forming bacteria and many fermentative organisms, and the Cyanobacteria, which perform oxygenic photosynthesis and were responsible for oxygenating Earth's atmosphere. Archaeal diversity reveals a group of organisms that superficially resemble bacteria, but exhibit fundamental differences in cellular components and biochemistry that reflect their distinct evolutionary heritage. Archaea include methanogens that produce methane in anaerobic environments, extreme halophils that thrive in hypersaline conditions, and thermophiles that inhabit high-temperature environments, though recent discoveries have revealed mesophilic archaea in diverse environments including soils and marine systems. Viral diversity encompasses an enormous variety of genetic and structural types that infect virtually every form of cellular life, from the simplest bacteria to complex eukaryotic organisms. Viruses exhibit remarkable diversity in genome organization, replication strategies, and host specificity, with recent metagenomic studies revealing that viral diversity far exceeds previous estimates, and that viruses play crucial roles in regulating microbial communities and driving evolution. Fungal diversity includes yeasts, molds, and other forms that exhibit diverse nutritional strategies and ecological roles, from decomposers that recycle organic matter to symbionts that form beneficial associations with plants and other organisms. Fungi demonstrate unique cellular and biochemical features, including chitin cell walls, high full growth forms, and specialized reproductive structures that enable them to exploit diverse environmental niches. Protist diversity encompasses a vast array of single-celled eukaryotic organisms that exhibit enormous variety in cellular organization, nutritional strategies, and ecological roles. These organisms include photosynthetic forms that serve as primary producers in aquatic environments, predatory forms that consume bacteria and other microorganisms, and parasitic forms that cause diseases in plants, animals, and other protists. Microbial biogeography examines the distribution patterns of microorganisms across space and time, revealing that microbial communities show distinct geographic patterns despite the potential for global dispersal through atmospheric transport and other mechanisms. These patterns reflect the influence of environmental conditions, evolutionary history, and ecological interactions that shape microbial community composition at local and global scales. Microbial habitats encompass virtually every environment on Earth, from the familiar soil and aquatic systems to extreme environments such as hot springs, salt lakes, and deep subsurface environments. Each habitat presents unique challenges and opportunities that have driven the evolution of specialized microbial communities, adapted to specific environmental conditions and resource availability patterns. Soil microbiology reveals complex communities that play crucial roles in nutrient cycling, plant health, and ecosystem functioning through processes including decomposition, nitrogen fixation, and disease suppression. Soil microbial communities are influenced by factors including soil chemistry, plant cover, moisture levels, and human activities that collectively determine community composition and function. Aquatic microbiology encompasses the study of microorganisms in freshwater and marine environments, where they serve as primary producers, decomposers, and links in complex food webs that support higher trophic levels. Aquatic microbial communities are structured by factors including nutrient availability, light penetration, temperature, and predation pressure that create distinct community patterns at different depths and locations. Extreme environment microbiology reveals organisms that thrive in conditions previously thought to be incompatible with life, expanding our understanding of the limits of biological existence, while providing insights into early evolution and the potential for life in extraterrestrial environments. These studies have revealed active microbial communities in environments including deep ocean trenches, acidic hot springs, hypersaline lakes, and deep subsurface rocks. Microbial communities exhibit complex structures and interactions that create emergent properties not present in individual organisms, including enhanced resistance to environmental stresses, division of labor among different community members, and sophisticated communication systems that coordinate community responses to environmental changes. Understanding these community-level properties is crucial for predicting how microbial ecosystems will respond to environmental perturbations. Symbiotic relationships involving microorganisms are ubiquitous in nature and include mutualistic associations that benefit all partners, commensalistic relationships where one partner benefits without affecting the other, and parasitic interactions where one partner benefits at the expense of the other. These relationships often involve intimate physiological integration and can be essential for the survival and functioning of both microbial and host organisms. Microbial succession describes the predictable changes in community composition that occur over time in response to environmental changes or during ecosystem development. These patterns reflect the changing availability of resources, the modification of environmental conditions by early colonizers, and competitive interactions that determine which organisms can establish and persist in different stages of succession. Horizontal gene transfer enables microorganisms to acquire genetic material from other organisms through mechanisms including transformation, transduction, and conjugation, creating opportunities for rapid adaptation and the spread of beneficial traits through microbial communities. This process is particularly important for the spread of antibiotic resistance and the acquisition of virulence factors in pathogenic organisms. Microbial loop describes the complex network of interactions involving bacteria, viruses, and protists that process dissolved organic matter in aquatic environments, demonstrating how microbial communities can form sophisticated food webs that operate largely independent of larger organisms. This process is crucial for nutrient cycling and energy flow in aquatic ecosystems. Biogeochemical cycling involves microbial processes that transform elements including carbon, nitrogen, sulfur, and phosphorus between different chemical forms, enabling the recycling of essential nutrients and maintaining the chemical balance necessary for ecosystem functioning. These processes operate at scales ranging from individual cells to global systems and are essential for maintaining environmental conditions compatible with life. Chapter 4: Pathogenic Microorganisms and Disease. Pathogenic microorganisms represent a small but medically significant fraction of microbial diversity, having evolved sophisticated mechanisms to invade, colonize, and exploit host organisms, while often causing disease processes that can range from mild discomfort to life-threatening conditions. Understanding the biology of pathogenic microorganisms and their interactions with hosts is crucial for developing effective strategies to prevent, diagnose, and treat infectious diseases that continue to pose significant challenges to human health worldwide. Bacterial pathogens encompass diverse species that cause diseases through various mechanisms, including toxin production, tissue invasion, immune system evasion, and disruption of normal physiological processes. These organisms have evolved specific virulence factors that enable them to overcome host defenses and establish infections, while their ability to acquire antibiotic resistance through genetic mechanisms poses increasing challenges for medical treatment. Virulence factors represent the molecular tools that enable pathogenic microorganisms to cause disease, including adhesins that facilitate attachment to host cells, toxins that damage host tissues or disrupt cellular functions, enzymes that break down host barriers, and immune evasion mechanisms that prevent effective host responses. The coordinated expression of these factors often determines the severity and progression of infectious diseases. Gram-positive bacterial pathogens include important species such as Streptococcus pyogenes, which causes strep throat and more serious invasive infections, Staphylococcus aureus, which causes skin infections and potentially fatal systemic diseases, and Clostridium difficile, which causes antibiotic-associated colitis. These organisms demonstrate diverse pathogenic strategies while sharing the common feature of thick peptidoglycan cell walls that influence their antibiotic susceptibility patterns. Gram-negative bacterial pathogens such as Escherichia coli, Salmonella species, and Pseudomonas aeruginosa possess outer membrane structures that provide additional protection against host defenses and many antibiotics, while often containing lipopolysaccharide components that can trigger severe inflammatory responses. These organisms are often associated with hospital-acquired infections and show increasing resistance to multiple antibiotics. Mycobacterial infections include tuberculosis and leprosy, caused by organisms with unique cell wall compositions that enable them to survive within host immune cells, while causing chronic infections that can persist for years or decades. These slow-growing organisms require specialized diagnostic and treatment approaches, while posing significant public health challenges, particularly in developing countries and immunocompromised populations. Viral pathogens demonstrate remarkable diversity in their replication strategies, host specificity, and disease-causing mechanisms, ranging from acute infections that resolve quickly to chronic infections that persist for the lifetime of the host. Understanding viral pathogenesis requires knowledge of how viruses attach to and enter host cells, replicate their genetic material, assemble new viral particles, and spread to new hosts. DNA viruses include important pathogens such as herpesviruses, which can establish latent infections that periodically reactivate, and papillomaviruses, which can cause both benign and malignant lesions. These viruses often integrate their genetic material into host chromosomes, or establish persistent infections that can influence host cell behavior and potentially contribute to cancer development. RNA viruses encompass a diverse group including influenza viruses, which undergo frequent genetic changes that necessitate annual vaccine updates, and retroviruses such as HIV, which integrate their genetic material into host chromosomes and establish persistent infections. The high mutation rates of many RNA viruses enable rapid evolution and adaptation to host immune responses and antiviral drugs. Emerging viral diseases continue to pose threats to global health through the spillover of viruses from animal reservoirs to human populations, often facilitated by environmental changes, increased human-animal contact, and global travel patterns. Recent examples include SARS-CoV-2, which caused the COVID-19 pandemic, and various influenza strains that periodically cause global outbreaks with significant morbidity and mortality. Fungal pathogens primarily affect immunocompromised individuals, though some species can cause infections in healthy hosts, particularly through respiratory exposure to spores or direct contact with contaminated materials. These organisms often produce chronic infections that are difficult to diagnose and treat, due to the similarity between fungal and human cellular components that limits the availability of selective antifungal agents. Parasitic microorganisms include protists such as Plasmodium species that cause malaria, Giardia that causes intestinal infections, and Toxoplasma that can cause serious infections in immunocompromised individuals and developing fetuses. These organisms often have complex life cycles involving multiple hosts and developmental stages that complicate treatment and prevention efforts. Host-pathogen interactions involve complex molecular and cellular processes that determine the outcome of microbial encounters with potential hosts, including the initial recognition and attachment events, the host immune response, and the pathogen's counter-responses that may enable infection establishment. Understanding these interactions is crucial for developing new therapeutic approaches and predicting the evolution of pathogenic organisms. Immune evasion strategies employed by pathogenic microorganisms include antigenic variation that enables escape from host antibody responses, molecular mimicry that prevents immune recognition, and direct interference with immune cell functions that disables host defenses. These strategies often involve sophisticated molecular mechanisms that reflect the evolutionary arms race between pathogens and host immune systems. Antibiotic resistance represents one of the most serious threats to modern medicine, with many pathogenic bacteria acquiring resistance to multiple antibiotics through mechanisms including enzymatic degradation of antibiotics, modification of antibiotic targets, and active efflux of antibiotics from bacterial cells. The spread of resistance through horizontal gene transfer and the selection pressure created by antibiotic use have created organisms that are resistant to virtually all available antibiotics. Antimicrobial stewardship involves coordinated efforts to optimize antibiotic use in order to minimize the development and spread of antibiotic resistance, while maintaining effective treatment options for infectious diseases. These programs include guidelines for appropriate antibiotic selection, duration of treatment, and infection prevention measures that can reduce the overall burden of antibiotic-resistant infections. Diagnostic microbiology has evolved from traditional culture-based methods to sophisticated molecular techniques that can rapidly identify pathogenic microorganisms and their antimicrobial susceptibility patterns. These advances enable more targeted therapy and improved patient outcomes while reducing the inappropriate use of broad-spectrum antibiotics that can contribute to resistance development. Epidemiology of infectious diseases involves the study of disease patterns in populations, including the identification of risk factors, transmission routes, and control measures that can prevent disease spread. Modern epidemiological investigations often incorporate molecular typing methods that can track the spread of specific pathogenic strains and identify the sources of outbreaks. One Health approaches recognize the interconnected nature of human, animal, and environmental health in the emergence and spread of infectious diseases, particularly those caused by zoonotic pathogens that can be transmitted between animals and humans. These approaches emphasize the need for coordinated surveillance and control measures that address the complex ecological factors influencing disease emergence. Public health microbiology involves the application of microbiological knowledge to protect population health through disease surveillance, outbreak investigation, vaccine development, and the implementation of control measures that can prevent the spread of infectious diseases. These efforts require coordination between laboratory scientists, clinicians, and public health officials to address emerging threats and maintain preparedness for future outbreaks. Chapter 5: Applied Microbiology and Biotechnology. Applied microbiology harnesses the extraordinary metabolic capabilities and rapid growth rates of microorganisms to address human needs and challenges, creating industries worth billions of dollars while providing sustainable solutions to problems ranging from environmental cleanup to pharmaceutical production. This field demonstrates how fundamental understanding of microbial biology can be translated into practical applications that benefit society while opening new frontiers for technological innovation. Industrial microbiology exploits the metabolic versatility of microorganisms to produce a vast array of commercially important compounds, including pharmaceuticals, enzymes, organic acids, amino acids, and vitamins through controlled fermentation processes. These applications take advantage of the ability of microorganisms to convert inexpensive raw materials into valuable products, while operating under conditions that would be impossible or economically unfavorable for chemical synthesis. Pharmaceutical production through microbial fermentation includes the manufacture of antibiotics, vaccines, hormones, and other therapeutic compounds that have revolutionized modern medicine. The production of penicillin by Penicillium fungi represents one of the earliest and most successful examples of pharmaceutical microbiology, while modern biotechnology enables the production of complex human proteins such as insulin and growth hormone using genetically modified microorganisms. Enzyme production represents a major application of industrial microbiology, with microorganisms serving as sources of enzymes used in food processing, textile manufacturing, paper production, and other industrial processes. These microbial enzymes often exhibit superior properties compared to enzymes from other sources, including higher stability, activity under extreme conditions, and specificity for particular substrates. Fermentation technology involves the controlled cultivation of microorganisms in bioreactors designed to optimize growth conditions and product formation, while maintaining sterility and enabling efficient product recovery. Modern fermentation systems incorporate sophisticated monitoring and control systems that can maintain optimal conditions for microbial growth and metabolism, while maximizing product yields and quality. Food microbiology encompasses both the beneficial roles of microorganisms in food production and preservation, as well as the challenges posed by food spoilage and foodborne pathogens. Beneficial applications include the production of fermented foods such as bread, cheese, yogurt, and alcoholic beverages, while food safety applications focus on preventing contamination and controlling pathogenic microorganisms that can cause foodborne illness. Fermented foods represent one of humanity's oldest applications of microbiology, with traditional fermentation processes enabling food preservation while creating products with enhanced flavors, nutritional value, and digestibility. These processes involve complex microbial communities that transform raw materials through coordinated metabolic activities that produce characteristic flavors, textures, and preservation effects. Food preservation techniques exploit antimicrobial properties of certain microorganisms or their metabolic products to prevent spoilage and extend shelf life, including the use of lactic acid bacteria to produce organic acids that inhibit pathogenic and spoilage organisms. Understanding the factors that control microbial growth in foods enables the development of preservation strategies that maintain safety and quality while minimizing the use of chemical preservatives. Environmental microbiology addresses the roles of microorganisms in environmental processes and their applications for environmental remediation, waste treatment, and pollution control. These applications exploit the ability of microorganisms to degrade pollutants, remove toxic compounds, and restore contaminated environments through natural metabolic processes that can be enhanced and directed through engineering approaches. Bioremediation uses microorganisms to remove or neutralize environmental contaminants through natural metabolic processes that can be enhanced through the addition of nutrients, electron acceptors, or specific microbial inoculants. These approaches have been successfully applied to clean up oil spills, remove heavy metals from contaminated soils, and treat groundwater contaminated with organic solvents and other industrial chemicals. Wastewater treatment relies heavily on microbial processes to remove organic matter, nutrients, and pathogens from domestic and industrial wastewater through activated sludge systems, biofilm reactors, and constructed wetlands that harness natural microbial communities. These systems demonstrate how understanding microbial ecology can be applied to design efficient and sustainable treatment processes. Agricultural microbiology explores the beneficial roles of microorganisms in plant growth and health, including nitrogen-fixing bacteria that provide nutrients to plants, mycorrhizal fungi that enhance nutrient uptake, and biocontrol agents that suppress plant pathogens. These applications offer sustainable alternatives to chemical fertilizers and pesticides, while improving crop productivity and environmental quality. Genetic engineering of microorganisms has revolutionized biotechnology by enabling the modification of microbial genomes to produce novel compounds, enhance existing capabilities, or create organisms with entirely new functions. These techniques include the insertion of foreign genes, the modification of existing genes, and the construction of synthetic biological circuits that can program microorganisms to perform specific tasks. Recombinant DNA technology enables the production of human and other eukaryotic proteins in bacterial hosts through the insertion of cloned genes into bacterial plasmids, creating opportunities to produce therapeutic proteins that would be impossible to obtain from natural sources. This technology has enabled the production of insulin, growth hormone, clotting factors, and numerous other therapeutic proteins that have transformed medical treatment. Metabolic engineering involves the systematic modification of microbial metabolic pathways to enhance the production of desired compounds or enable the synthesis of novel products through the optimization of enzyme activities, elimination of competing pathways, and introduction of new metabolic capabilities. These approaches are being applied to produce biofuels, pharmaceuticals, and industrial chemicals from renewable feedstocks. Synthetic biology represents an emerging field that applies engineering principles to biological systems through the design and construction of novel biological parts, devices, and systems that can perform specific functions. This approach involves the standardization of biological components and the development of design principles that can guide the construction of predictable and reliable biological systems. Biofuel production exploits the ability of microorganisms to convert biomass into fuels such as ethanol, biodiesel, and biogas through fermentation and other metabolic processes. These applications offer renewable alternatives to fossil fuels while creating opportunities to convert agricultural and municipal wastes into valuable energy products. Microbial fuel cells harness the ability of certain microorganisms to generate electrical current through their metabolic activities, creating opportunities for sustainable energy production while treating organic wastes. These systems demonstrate how microbial metabolism can be coupled to electrical circuits to generate power from organic matter in wastewater and other waste streams. Biosensors incorporate microorganisms or microbial components into devices that can detect specific chemicals or conditions through biological recognition mechanisms, coupled to electronic signal transduction systems. These devices offer sensitive and specific detection capabilities for environmental monitoring, medical diagnostics, and industrial process control applications. Quality control in applied microbiology involves sophisticated testing and monitoring procedures that ensure the safety, efficacy, and consistency of microbial products and processes. These procedures include sterility testing, endotoxin detection, identity confirmation, and potency assays that are essential for maintaining product quality and regulatory compliance. Regulatory considerations for microbial products and processes involve complex frameworks that balance innovation with safety and environmental protection through requirements for extensive testing, documentation, and oversight of microbial applications. Understanding these regulatory requirements is essential for successful commercialization of microbial technologies, while ensuring public safety and environmental protection. Future directions in applied microbiology include the development of more sophisticated genetic tools, the exploration of novel microbial capabilities, and the integration of microbiology with other disciplines to address complex challenges including climate change, sustainable development, and human health. These advances promise to expand the applications of microbiology while creating new opportunities for innovation and discovery.
Chapter 1: Foundations of Plant Ecology. Plant ecology represents one of the most fundamental branches of biological science, examining the intricate relationships between plants and their environment while revealing the complex web of interactions that sustain life on Earth. At its essence, plant ecology seeks to understand how vegetation responds to environmental conditions, how plant communities assemble and change over time, and how these green organisms shape the very ecosystems they inhabit. This field of study encompasses everything from the molecular mechanisms that allow plants to sense and respond to their surroundings, to the vast landscape-scale patterns of vegetation that define our planet's biomes. The history of plant ecology stretches back centuries, with early naturalists like Alexander von Humboldt recognizing that plant distributions followed predictable patterns related to climate and geography. Humboldt's observations in the early 1800s laid the groundwork for understanding vegetation zones and the relationship between plant communities and environmental gradients. His work established the fundamental principle that plants are not randomly distributed across landscapes, but instead form predictable assemblages based on environmental conditions such as temperature, precipitation, soil characteristics, and elevation. The development of plant ecology as a formal scientific discipline accelerated during the late 19th and early 20th centuries, driven by pioneering researchers who established many of the field's core concepts. Frederick Clements proposed his influential theory of ecological succession, suggesting that plant communities develop through predictable stages toward a stable climax state determined by climate. While later research has revealed the limitations of this linear view of succession, Clements' work established the importance of temporal dynamics in plant community development and highlighted the interconnected nature of vegetation change. Henry Gleason offered an alternative perspective through his individualistic concept of plant communities, arguing that plant distributions result from the independent responses of individual species to environmental conditions rather than highly integrated community-level processes. This debate between organismic and individualistic views of plant communities continues to influence ecological thinking today, with modern research revealing elements of truth in both perspectives depending on the spatial and temporal scales of investigation. Modern plant ecology has evolved into a sophisticated science that integrates multiple approaches and scales of investigation. Physiological ecology examines how individual plants function and respond to environmental stresses, revealing the mechanisms underlying plant adaptation and survival. Population ecology focuses on the dynamics of single species, investigating factors that control birth, death, and reproduction rates. Community ecology explores interactions among multiple species, including competition, facilitation, and the complex networks of relationships that structure plant assemblages. The emergence of ecosystem ecology has broadened the scope of plant ecological research to encompass energy flows, nutrient cycling, and the role of vegetation in biogeochemical processes. This systems-level approach recognizes that plants are not merely passive recipients of environmental conditions, but active agents that modify their surroundings through processes such as photosynthesis, transpiration, and litter production. Plants alter soil chemistry, influence local climate conditions, and create habitat heterogeneity that affects the distribution and abundance of other organisms. Landscape ecology has further expanded the spatial scope of plant ecological research, examining how vegetation patterns and processes vary across heterogeneous landscapes. This perspective recognizes that plant communities exist within a matrix of patches connected by flows of organisms, materials, and energy. Understanding these landscape-scale patterns and processes has become increasingly important for conservation biology and land management, particularly as human activities continue to fragment natural habitats and alter ecosystem connectivity. The tools and methods available to plant ecologists have undergone revolutionary changes in recent decades, driven by advances in technology and analytical techniques. Remote sensing technologies now allow researchers to monitor vegetation patterns and changes across vast spatial scales and extended time periods. Satellite imagery and aerial photography provide detailed information about vegetation structure, productivity, and responses to disturbance events such as fires, droughts, and human land use changes. Molecular techniques have opened new frontiers in plant ecological research, enabling investigations of plant microbe interactions, genetic diversity within and among populations, and the evolutionary history of plant communities. Environmental DNA sampling can reveal the presence of rare or cryptic species, while isotopic analyses provide insights into plant water and nutrient sources, carbon allocation patterns, and historical environmental conditions preserved in plant tissues. Experimental approaches in plant ecology range from controlled laboratory studies that isolate specific environmental factors to large-scale field experiments that manipulate entire ecosystems. Long-term ecological research sites provide invaluable data on vegetation dynamics over multiple decades, revealing patterns and processes that operate on timescales far exceeding typical research projects. These long-term datasets have been instrumental in documenting ecosystem responses to climate change, atmospheric pollution, and other global environmental changes. The theoretical framework of plant ecology continues to evolve as new concepts and models are developed to explain observed patterns and predict future changes. Neutral theory challenges traditional niche-based explanations of community structure by proposing that stochastic processes and random dispersal may explain many patterns previously attributed to deterministic ecological interactions. Metacommunity theory incorporates spatial dynamics and dispersal limitation into models of community assembly and maintenance, recognizing that local communities are connected through regional species pools and migration processes. Plant functional ecology has emerged as a unifying framework that focuses on plant traits and their relationships to environmental conditions and ecological processes. This approach recognizes that species with similar functional characteristics may play similar ecological roles regardless of their evolutionary relationships. Functional traits such as leaf area, wood density, seed size, and root architecture provide mechanistic links between environmental conditions and plant performance, enabling predictions about how vegetation will respond to environmental change. The integration of plant ecology with other disciplines has yielded rich insights into the functioning of Earth systems. Collaborations with atmospheric scientists have revealed the crucial role of vegetation in regulating climate through carbon sequestration, albedo effects, and the emission of volatile organic compounds. Partnerships with hydrologists have illuminated how vegetation influences water cycling through transpiration, interception, and modification of soil hydraulic properties. Interactions with soil scientists have enhanced understanding of plant-soil feedbacks and the role of vegetation in ecosystem nutrient cycling. Plant ecology has also become increasingly applied, with research findings directly informing conservation strategies, restoration practices, and sustainable land management. Understanding plant community assembly and succession dynamics guides restoration efforts aimed at re-establishing native vegetation on degraded lands. Knowledge of plant responses to environmental stresses informs strategies for conserving rare species and managing protected areas. Insights into plant-herbivore interactions and invasive species ecology support pest management and biological control programs. The field faces numerous challenges and opportunities as global environmental changes accelerate and human impacts on natural systems intensify. Climate change is altering temperature and precipitation patterns worldwide, forcing rapid responses from plant communities and creating novel environmental conditions that may favor new species assemblages. Rising atmospheric carbon dioxide concentrations are directly affecting plant physiology and growth, with cascading effects on community structure and ecosystem processes. Habitat fragmentation and land use change continue to threaten plant diversity and alter ecological processes across landscapes. Understanding how plant communities respond to these changes and identifying strategies for maintaining ecosystem integrity in human-dominated landscapes represents a critical challenge for contemporary plant ecology. Urban ecology has emerged as an important subdiscipline focused on plant communities in cities and suburban areas, where novel environmental conditions and management practices create unique ecological challenges and opportunities. Chapter 2: Plant Adaptations and Environmental Responses. Plants have evolved remarkable adaptations that enable them to survive and thrive across Earth's diverse environments, from the frozen soils of Arctic tundra to the scorching heat of desert landscapes, from the nutrient-poor conditions of bog ecosystems to the salt-laden environments of coastal marshes. These adaptations represent millions of years of evolutionary innovation, resulting in sophisticated physiological, morphological, and biochemical mechanisms that allow plants to cope with environmental challenges while competing effectively for limited resources such as light, water, and nutrients. Water availability represents one of the most fundamental constraints on plant survival and distribution, driving the evolution of diverse strategies for water acquisition, conservation, and management. Plants in arid environments have developed numerous xeromorphic adaptations that minimize water loss while maximizing water uptake and storage. Succulents such as cacti and agaves possess thick, waxy stems and leaves that store large quantities of water during brief periods of rainfall, while their specialized crassulation acid metabolism allows them to open their stomata at night when evaporation rates are lower, reducing water loss during photosynthesis. Desert shrubs often exhibit small, thick leaves with extensive waxy coatings and dense pubescence that reflect solar radiation and create boundary layers of still air that reduce transpiration. Many desert plants have evolved extensive root systems that either penetrate deeply to access groundwater or spread widely near the surface to capture precipitation from infrequent rainfall events. Some species, such as desert willow and mesquite, combine both strategies with dimorphic root systems that include both deep taproots and shallow lateral roots. In contrast, plants in water-saturated environments face the challenge of obtaining adequate oxygen for root respiration while preventing the accumulation of toxic compounds that develop under anaerobic conditions. Wetland plants have evolved specialized anatomical structures called parenchyma, which consist of large air-filled spaces in roots and stems that facilitate oxygen transport from aerial portions of the plant to submerged roots. Mangroves, which inhabit saltwater environments, have developed pneumatophores or aerial roots that protrude above the water surface to access atmospheric oxygen. Temperature extremes have driven the evolution of numerous plant adaptations that enable survival in both extremely hot and cold environments. Plants in cold climates often exhibit morphological features that help them conserve heat and protect sensitive tissues from freezing damage. Alpine and Arctic plants frequently grow in cushion or rosette forms that create favorable microclimates by trapping warm air and reducing wind exposure. These growth forms also position reproductive structures close to the warm soil surface, extending the growing season and improving reproductive success. Cold-adapted plants have evolved sophisticated biochemical mechanisms to prevent cellular damage from freezing temperatures. Many species accumulate compatible solutes, such as sugars, amino acids, and specialized proteins that act as antifreeze compounds, lowering the freezing point of cellular fluids and protecting cell membranes from ice crystal formation. Some plants undergo cold hardening processes that gradually increase their freezing tolerance as temperatures decline, allowing them to survive progressively colder conditions throughout the winter months. Plants in extremely hot environments have developed complementary strategies for dealing with high temperatures and intense solar radiation. Heat-adapted plants often possess reflective surfaces created by specialized hairs, scales, or waxy coatings that reduce the absorption of solar energy. Some species exhibit heliotropism, the ability to track the sun's movement throughout the day, which can either maximize light capture during cooler periods or minimize exposure during the hottest parts of the day, depending on the species' adaptive strategy. The biochemistry of photosynthesis itself has evolved multiple times to optimize carbon fixation under different environmental conditions. C4 photosynthesis, found in many grass species and other plants from hot, dry environments, concentrates carbon dioxide around the enzyme rubisco, improving photosynthetic efficiency and reducing photorespiration under high temperature and light conditions. CAM photosynthesis represents an even more extreme adaptation to arid conditions, allowing plants to separate the temporal aspects of carbon dioxide uptake and fixation, thereby minimizing water loss during the hottest parts of the day. Light availability and quality create additional environmental challenges that have shaped plant evolution in profound ways. Forest understory plants must cope with low light conditions while competing for the brief periods of high light that occur when canopy gaps form. Many understory species have evolved large, thin leaves with high chlorophyll concentrations that maximize light capture efficiency under shaded conditions. Some plants exhibit remarkable phenotypic plasticity, dramatically altering their leaf characteristics in response to changing light conditions as forest canopies develop overhead. Shade-tolerant plants often invest heavily in light harvesting complexes and maintain low respiration rates that allow them to maintain positive carbon balance even under extremely low light conditions. In contrast, pioneer species that colonize disturbed areas typically exhibit characteristics that allow rapid growth under high light conditions, including high photosynthetic capacity, rapid resource acquisition, and efficient resource allocation to above-ground biomass. Nutrient availability, particularly nitrogen and phosphorus, represents another major environmental constraint that has driven plant adaptive evolution. Plants in nutrient-poor environments have evolved various strategies for maximizing nutrient acquisition and retention. Many species form symbiotic relationships with mycorrhizal fungi that greatly expand their effective root surface area and improve their ability to extract nutrients from soil. Ectomycorrhizal associations are particularly common among trees in temperate and boreal forests, while arbuscular mycorrhizal fungi form partnerships with many herbaceous plants and tropical trees. Some plants have evolved carnivorous adaptations that allow them to supplement their nutrient supply by capturing and digesting insects and other small animals. These adaptations are particularly common in nutrient-poor environments, such as bogs and sandy soils, where conventional root uptake cannot meet plant nutritional requirements. Carnivorous plants exhibit diverse mechanisms for prey capture, including pitfall traps, snap traps, sticky surfaces, and suction mechanisms, demonstrating the evolutionary flexibility of plant form and function. Leguminous plants have evolved symbiotic relationships with nitrogen-fixing bacteria that allow them to convert atmospheric nitrogen into forms that can be used for plant growth. This adaptation provides a significant competitive advantage in nitrogen-limited environments and has allowed legumes to colonize disturbed soils and serve as pioneer species in many ecosystems. The ability to fix nitrogen also makes legumes important components of sustainable agricultural systems and ecosystem restoration efforts. Soil chemistry and pH present additional challenges that have shaped plant distributions and evolutionary adaptations. Plants growing on calcareous soils must cope with high pH conditions and potential micronutrient deficiencies, particularly iron chlorosis. Calcifugous plants have evolved mechanisms for acidifying their rhizosphere and mobilizing nutrients under alkaline conditions. Conversely, plants on acidic soils must tolerate high concentrations of aluminum and other potentially toxic metals while coping with reduced availability of essential nutrients such as phosphorus. Some plants have evolved remarkable tolerance to heavy metals and other soil toxins, allowing them to colonize contaminated sites that are unsuitable for most vegetation. These metallophytes have developed mechanisms for excluding toxic metals from sensitive tissues, sequestering metals in specialized cellular compartments, or even accumulating metals to concentrations that would be lethal to other plants. Hyperaccumulator plants that concentrate metals in their tissues have become important tools for phytoremediation of contaminated soils. Salt tolerance represents another specialized adaptation that allows plants to colonize saline environments such as salt marshes, alkaline flats, and coastal areas. Halophytic plants have evolved diverse mechanisms for coping with high salt concentrations, including salt exclusion at the root level, compartmentalization of salts in specialized tissues or cellular organelles, and the use of compatible solutes to maintain cellular water balance. Some halophytes actually require salt for optimal growth and cannot survive in freshwater environments. The temporal dimension of environmental variation has also driven important plant adaptations, particularly in seasonal environments where resource availability and environmental conditions change predictably throughout the year. Deciduous trees shed their leaves before winter to avoid damage from freezing temperatures and to conserve water when soil water may be frozen and unavailable. The timing of leaf drop and bud break represents a critical adaptation that must balance the benefits of extending the growing season against the risks of frost damage. Phenological adaptations that synchronize plant activities with seasonal environmental cycles are crucial for reproductive success and survival. Many temperate plants require specific chilling requirements before they can break dormancy in spring, ensuring that growth does not begin during temporary warm spells that might be followed by damaging frosts. Desert plants often exhibit drought deciduous habits, dropping leaves during dry periods and rapidly producing new foliage when moisture becomes available. Chapter 3: Plant Communities and Interactions. Plant communities represent complex assemblages of species that coexist within defined spatial boundaries, creating intricate networks of interactions that shape ecosystem structure, function, and dynamics. Understanding how these communities form, persist, and change over time requires examination of the diverse mechanisms through which plants interact with one another, ranging from intense competition for limited resources to mutually beneficial relationships that enhance the survival and reproduction of participating species. The study of plant community ecology reveals that vegetation patterns emerge from the interplay between environmental filtering, species interactions, and historical contingencies that operate across multiple spatial and temporal scales. Competition represents one of the most pervasive and well-studied forms of interaction among plants, occurring whenever individuals attempt to utilize the same limited resources. Above-ground competition for light creates some of the most dramatic examples of plant competitive interactions, driving the evolution of diverse strategies for light capture and canopy positioning. In forest ecosystems, the vertical structure of vegetation reflects intense competition for photosynthetically active radiation, with canopy trees investing heavily in height growth and structural support to position their leaves above competitors. The process of canopy development involves complex feedback loops between individual plant growth and community-level light environments. As some individuals gain height advantages, they cast increasing shade on their neighbors, creating positive feedback loops that can lead to competitive exclusion of shorter individuals. However, the formation of canopy gaps through tree mortality creates opportunities for understory plants and new recruits, maintaining diversity within forest communities through spatial and temporal heterogeneity in light availability. Below-ground competition for water and nutrients presents challenges that are often less visible but equally important for plant community dynamics. Root systems exhibit remarkable plasticity in response to soil resource heterogeneity, with plants capable of rapidly proliferating roots in nutrient-rich patches while reducing investment in areas with poor resource availability. This foraging behavior can lead to intense local competition as multiple plants attempt to exploit the same resource patches. The intensity of competitive interactions varies dramatically among different types of plant communities and environmental conditions. In productive environments where resources are abundant, competition tends to be primarily for light, leading to selection for rapid height growth and high leaf area indices. In contrast, competition in resource-poor environments often focuses on below-ground resources, favoring plants with extensive root systems and efficient nutrient uptake mechanisms. Facilitation represents the opposite end of the interaction spectrum from competition, encompassing situations where the presence of one plant species enhances the survival, growth, or reproduction of another species. These positive interactions are particularly common in stressful environments, where amelioration of harsh conditions provides significant benefits to neighboring plants. In arid ecosystems, nurse plants often provide shade, improved soil conditions, and protection from herbivores that benefit other species, creating islands of enhanced diversity around facilitating individuals. Alpine and Arctic environments provide numerous examples of facilitative interactions, where cushion plants and other stress-tolerant species create favorable microclimates that enable less hardy species to survive at elevations or latitudes beyond their normal ranges. These facilitating species often modify soil temperature, reduce wind exposure, and improve moisture retention, creating positive feedback loops that enhance local diversity and productivity. The balance between competition and facilitation can shift along environmental gradients, with facilitation becoming more important under stressful conditions while competition dominates in benign environments. This stress gradient hypothesis has been supported by research across diverse ecosystems, revealing that the net effects of plant interactions depend critically on environmental context. Understanding these shifting interaction patterns is crucial for predicting how plant communities will respond to environmental change and for designing effective restoration strategies. Allelopathy represents a specialized form of plant interaction involving the release of chemical compounds that influence the growth, survival, or reproduction of other plants. These biochemical interactions can either inhibit competitors through the production of toxic secondary metabolites or facilitate neighbors through the release of growth-promoting compounds. Allelopathic interactions are particularly well documented in agricultural systems, where crop-weed interactions often involve chemical warfare between species. Many invasive plant species employ allelopathic strategies to establish dominance in their new environments, producing novel chemical compounds that native species have not evolved defenses against. Black walnut trees provide a classic example of allelopathy, producing juglone and other compounds that inhibit the growth of many understory species, creating characteristic zones of reduced diversity around individual trees. Plant-pollinator interactions represent crucial mutualisms that link plant communities with animal populations and influence patterns of gene flow, reproduction, and community assembly. The diversity and structure of plant communities often reflect the availability and efficiency of pollinator services, with specialized pollination systems creating tight linkages between particular plant and animal species. These relationships can create complex networks of interdependence that influence the stability and resilience of entire communities. The timing of flowering within plant communities reflects both resource availability and the need to attract pollinators, while avoiding excessive competition for pollinator services. Many communities exhibit sequential flowering patterns that spread pollinator demand across the growing season, reducing interspecific competition while maintaining consistent resource availability for pollinator populations. These temporal patterns can be disrupted by environmental changes that alter flowering phenology, potentially leading to mismatches between plants and their pollinators. Seed dispersal interactions similarly link plant communities with animal dispersers and influence patterns of colonization, gene flow, and community assembly. The structure of seed dispersal networks can determine which species are able to colonize new habitats and how rapidly plant communities can respond to environmental changes or disturbance events. Long-distance dispersal events, though rare, can be particularly important for maintaining connectivity among fragmented populations and enabling range shifts in response to climate change. Plant-herbivore interactions represent another fundamental force shaping plant community structure and dynamics. Herbivora can act as a selective pressure, favoring the evolution of defensive traits such as thorns, toxic secondary compounds, or tolerance mechanisms that allow plants to regrow after tissue removal. The intensity and selectivity of herbivora can dramatically alter competitive relationships among plants, sometimes allowing less competitive species to persist by reducing the dominance of superior competitors. Large herbivores can create and maintain particular vegetation types through their selective feeding and trampling activities. Grazing systems often exhibit alternative stable states, with the same environmental conditions potentially supporting either grassland or woodland vegetation, depending on herbivore pressure and disturbance history. Understanding these herbivore-mediated transitions is crucial for rangeland management and conservation of grassland ecosystems. The role of plant pathogens in community ecology has received increasing attention as researchers recognize the importance of species-specific natural enemies in maintaining plant diversity. The Janssen-Connell hypothesis proposes that specialized pathogens prevent any single plant species from becoming overwhelmingly dominant by creating density-dependent mortality that maintains space for other species. This mechanism may be particularly important in diverse tropical forests, where high pathogen diversity creates strong pressure against monospecific stands. Soil microbial communities play increasingly recognized roles in plant community dynamics through their effects on nutrient cycling, plant-soil feedbacks, and pathogen dynamics. Plants actively modify their rhizosphere communities through root exudates and other mechanisms, creating species-specific soil environments that can either favor or inhibit subsequent plant growth. These plant-soil feedbacks can influence succession dynamics, invasion susceptibility, and the coexistence of competing species. The spatial arrangement of plants within communities influences interaction strengths and community dynamics through effects on resource competition, microclimate modification, and the spread of pathogens or beneficial organisms. Spatial patterns can emerge from limited dispersal, environmental heterogeneity, or positive and negative interactions among individuals. Understanding these spatial dynamics is crucial for predicting community responses to fragmentation, climate change, and other environmental pressures. Scale dependence represents a fundamental characteristic of plant community interactions, with different processes dominating at different spatial and temporal scales. Local interactions among neighboring individuals may be overwhelmed by regional processes such as climate variation, disturbance events, or dispersal limitation. Metacommunity theory provides a framework for understanding how local communities are embedded within regional species pools and connected through dispersal and colonization processes. Chapter 4: Succession and Ecosystem Dynamics. Ecological succession represents one of the most fundamental processes in plant ecology, describing the predictable changes in vegetation composition and structure that occur following disturbance or during the colonization of new habitats. This dynamic process reveals how plant communities develop over time through complex interactions among colonization, competition, facilitation, and environmental modification, ultimately leading to the assembly of mature ecosystems with characteristic species compositions and ecological functions. Understanding succession provides crucial insights into ecosystem resilience, restoration ecology, and the long-term dynamics of vegetation in response to both natural and anthropogenic disturbances. Primary succession occurs in previously unvegetated environments where soil development must accompany plant colonization, such as newly exposed volcanic surfaces, glacial moraines, or sand dunes. These harsh environments present extreme challenges for plant establishment, including unstable substrates, extreme temperatures, high solar radiation, and minimal nutrient availability. Pioneer species that successfully colonize these environments typically possess specialized adaptations for stress tolerance, including the ability to fix nitrogen, tolerate low nutrient conditions, and establish rapidly from small propagules dispersed over long distances. Lichens and mosses often serve as the initial colonizers of bare rock surfaces, beginning the slow process of soil formation through physical and chemical weathering of the underlying substrate. These cryptobiotic communities can survive extreme environmental conditions and gradually accumulate organic matter and nutrients that facilitate the establishment of vascular plants. The transition from cryptobiotic crusts to grassland or shrubland vegetation may require decades or centuries, depending on climate conditions and the rate of soil development. The establishment of nitrogen-fixing species represents a crucial step in primary succession, as these plants can overcome nitrogen limitation that prevents the growth of other species. Alder species commonly serve as nitrogen-fixing pioneers in temperate regions, rapidly building soil nitrogen pools that support the establishment of other tree species. Similarly, leguminous shrubs and herbs often dominate early successional stages in many ecosystems, creating positive feedback loops that accelerate succession by improving soil fertility. Secondary succession occurs in areas where vegetation has been removed but soil remains intact, such as abandoned agricultural fields, logged forests, or areas recovering from fire or windstorms. Because soil development is not required, secondary succession typically proceeds more rapidly than primary succession, though the specific trajectory and rate of change depend on numerous factors including climate, soil properties, seed sources, and disturbance history. Old field succession provides some of the most thoroughly studied examples of secondary succession, revealing predictable patterns of species replacement as communities develop from annual herbs through perennial grasslands to shrublands and eventually forests. Early successional species typically exhibit characteristics that allow rapid colonization and growth under high light conditions, including high reproductive output, efficient dispersal mechanisms, and rapid growth rates. These R-selected species invest heavily in reproduction rather than competitive ability, allowing them to quickly exploit newly available resources. As succession proceeds, competitive interactions become increasingly important, favoring species with greater competitive ability for light, water, and nutrients. Mid-successional species often exhibit intermediate characteristics, combining moderate growth rates with greater competitive ability and stress tolerance. These species may persist for decades or longer, as ecosystems develop toward mature states dominated by K-selected species with high competitive ability, efficient resource use, and greater longevity. The classical view of succession as a predictable, linear progression toward a single climax state has been substantially modified by modern ecological research, which recognizes that successional trajectories can be highly variable and influenced by numerous contingent factors. Alternative stable states may exist under the same environmental conditions, with the specific trajectory determined by initial conditions, disturbance timing and intensity, dispersal limitation, and stochastic events that influence species establishment and survival. The intermediate disturbance hypothesis proposes that moderate levels of disturbance maintain maximum diversity by preventing competitive exclusion, while avoiding the severe impacts that would eliminate most species. This concept helps explain why many ecosystems exhibit complex mosaics of successional stages rather than uniform mature vegetation, as repeated disturbances create spatial and temporal heterogeneity that maintains diverse plant communities across landscapes. Disturbance characteristics, including frequency, intensity, extent, and predictability, profoundly influence successional dynamics and the resulting vegetation patterns. Fire regimes provide excellent examples of how disturbance properties shape ecosystem development, with different fire frequencies and intensities favoring different plant communities and successional trajectories. Frequent, low-intensity fires may maintain grassland or savanna vegetation by preventing tree establishment, while infrequent, high-intensity fires may reset succession to early stages dominated by pioneer species. Plant life history strategies have evolved in response to different disturbance regimes, creating predictable relationships between disturbance characteristics and plant community composition. Serotinous cones of some pine species require heat from fires to open and release seeds, ensuring that reproduction is synchronized with the creation of favorable establishment conditions. Resprouting ability allows many woody plants to rapidly reestablish after fire or other disturbances that remove above-ground biomass while leaving root systems intact. The seed bank represents a crucial component of successional dynamics, providing a reservoir of dormant propagules that can respond to disturbance events by germinating when conditions become favorable. Soil seed banks can contain viable seeds of species that have been absent from the vegetation for years or decades, allowing rapid recolonization following disturbance. The composition and longevity of seed banks vary among ecosystems and influence the potential for ecosystem recovery and restoration. Dispersal limitation often constrains successional dynamics, particularly in fragmented landscapes where seed sources may be distant from disturbed areas. The absence of key species during succession can lead to alternative successional trajectories or arrested development that prevents the formation of mature ecosystem states. Understanding dispersal limitation has become increasingly important for restoration ecology and conservation biology as human activities continue to fragment natural habitats. Facilitation plays particularly important roles during early succession when environmental conditions are harsh, and amelioration by established plants can dramatically improve conditions for other species. Nurse plants provide shade, wind protection, and improved soil conditions that benefit other species, creating positive feedback loops that accelerate succession. The death of facilitating species can trigger new phases of succession as the environmental conditions they created change. Ecosystem engineering by plants represents a fundamental mechanism through which vegetation modifies its environment and influences successional trajectories. Trees alter light environments, soil chemistry, and microclimate conditions in ways that favor shade-tolerant understory species while disadvantaging light-demanding pioneers. These environmental modifications create positive feedback loops that stabilize mature forest states and make reversal to earlier successional stages unlikely without major disturbance. Succession involves not only changes in species composition, but also fundamental shifts in ecosystem structure and function. Early successional ecosystems typically exhibit high primary productivity, rapid nutrient cycling, and simple food webs dominated by herbivora. Mature ecosystems often show lower net productivity, more complex nutrient cycling patterns with greater nutrient retention, and more complex food webs with greater importance of detrital pathways. The relationship between biodiversity and succession remains an active area of research, with different patterns observed at different taxonomic groups and spatial scales. Plant species diversity often peaks during intermediate stages of succession, when both early and late successional species coexist, though this pattern can vary depending on productivity, disturbance history, and regional species pools. Understanding these diversity patterns is crucial for conservation planning and ecosystem management. Climate change is altering successional dynamics worldwide by changing disturbance regimes, species ranges, and environmental conditions. Warmer temperatures may accelerate succession in some ecosystems while slowing it in others, depending on how temperature changes affect moisture availability, growing season length, and species interactions. Shifting precipitation patterns may favor different successional pathways or create novel environmental conditions that support unprecedented species assemblages. Human activities have fundamentally altered successional processes across much of the Earth's surface through habitat fragmentation, species introductions, pollution, and altered disturbance regimes. Many ecosystems now exist in permanently altered states that may never return to their historical conditions without intensive management intervention. Understanding these anthropogenic influences on succession is crucial for developing realistic conservation goals and effective restoration strategies. Restoration ecology applies successional principles to accelerate the recovery of degraded ecosystems, though this application reveals the complexity and contingency of successional processes. Successful restoration often requires careful attention to site preparation, species selection, establishment techniques, and long-term management to guide succession toward desired endpoints. The recognition that historical reference conditions may no longer be achievable or appropriate has led to greater emphasis on functional restoration and adaptation to changing environmental conditions. Chapter 5: Plant Ecology in a Changing World. The modern era presents unprecedented challenges to plant communities worldwide, as human activities fundamentally alter the environmental conditions that have shaped vegetation patterns for millennia. Climate change, habitat fragmentation, invasive species, pollution, and land use conversion are creating novel environmental conditions and ecological pressures that test the adaptive capacity of plant species and the resilience of ecosystems. Understanding how plant communities respond to these rapid changes and developing strategies to maintain ecosystem integrity under altered conditions represents one of the most urgent challenges facing contemporary plant ecology. Climate change manifests through multiple pathways that affect plant communities, including rising temperatures, altered precipitation patterns, increased frequency of extreme weather events, and elevated atmospheric carbon dioxide concentrations. These changes operate across multiple temporal and spatial scales, from immediate physiological responses to long-term evolutionary adaptations, creating complex challenges for predicting and managing ecosystem responses. Rising global temperatures are already shifting the geographic ranges of many plant species, with mountain and Arctic species facing particularly severe threats as suitable habitat moves upward in elevation or latitude faster than many species can migrate. Temperature increases affect plant communities through direct physiological impacts and indirect effects on water availability, growing season length, and species interactions. Many temperate plant species require specific chilling requirements for proper dormancy and flowering, and warmer winters may disrupt these processes, leading to reduced reproduction or increased vulnerability to frost damage. Heat stress during summer months can reduce photosynthetic efficiency and increase respiration rates, potentially altering competitive relationships among species with different thermal tolerances. Altered precipitation patterns present equally significant challenges for plant communities, particularly in regions where rainfall is becoming more variable or extreme. Prolonged droughts stress plant communities adapted to mesic conditions, potentially leading to widespread mortality and shifts toward more drought-tolerant vegetation. Conversely, increased precipitation in arid regions may favor invasive species or fundamentally alter ecosystem dynamics in areas where water has historically been limiting. The increased frequency and intensity of extreme weather events, including hurricanes, floods, droughts, and heat waves, create novel disturbance regimes that may exceed the adaptive capacity of existing plant communities. These extreme events can cause widespread mortality, alter competitive relationships, and create opportunities for species invasions or shifts in community composition. Recovery from extreme events may be increasingly difficult as environmental conditions continue to change during the recovery period. Elevated atmospheric carbon dioxide concentrations directly affect plant physiology through impacts on photosynthesis, water use efficiency, and plant nutrition. While increased carbon dioxide can enhance photosynthetic rates in many species, particularly those using C3 photosynthesis, these benefits are often limited by other factors such as nutrient availability or temperature stress. The carbon dioxide fertilization effect may also diminish over time as plants acclimate to elevated atmospheric concentrations, and the benefits tend to be greater for woody species than herbaceous plants, potentially altering competitive relationships within communities. Changes in atmospheric chemistry extend beyond carbon dioxide to include elevated nitrogen deposition, ozone pollution, and other atmospheric contaminants that affect plant communities. Nitrogen deposition can dramatically alter competitive relationships in nutrient-limited ecosystems, often favoring fast-growing species that can rapidly respond to increased nitrogen availability, while disadvantaging stress-tolerant species adapted to low nutrient conditions. These changes can lead to reduced plant diversity and altered ecosystem functioning. Habitat fragmentation represents another major driver of change in plant communities, breaking large continuous habitats into smaller isolated patches surrounded by altered landscapes. Fragmentation affects plant communities through multiple mechanisms, including edge effects that alter environmental conditions near fragment boundaries, reduced population sizes that increase extinction risk, and limited dispersal between fragments that reduces gene flow and colonization potential. Edge effects penetrate varying distances into habitat fragments, creating altered microclimatic conditions, increased exposure to invasive species, and modified disturbance regimes. These effects can fundamentally change species composition and ecosystem processes within fragments, often favoring generalist species adapted to variable conditions while disadvantaging specialist species requiring stable habitat conditions. The ratio of edge to interior habitat increases dramatically as fragments become smaller, potentially eliminating interior habitat conditions entirely in small fragments. Population sizes within fragments may fall below viable thresholds for long-term persistence, particularly for rare species or those with specialized habitat requirements. Small populations are vulnerable to demographic stochasticity, environmental fluctuations, and genetic drift that can lead to local extinctions. The loss of species from fragments can trigger cascading effects on remaining community members through altered competitive relationships, pollination networks, and seed dispersal systems. Invasive species represent one of the most pervasive threats to native plant communities worldwide, with human transportation networks facilitating the movement of species far beyond their natural ranges. Successful invaders often possess characteristics that allow rapid establishment and spread in novel environments, including high reproductive output, efficient dispersal mechanisms, broad environmental tolerance, and freedom from natural enemies that limit their populations in native ranges. The impacts of invasive species on native plant communities can be severe and long-lasting, often leading to reduced native diversity, altered ecosystem processes, and modified habitat conditions that favor further invasion. Invasive species may compete directly with native species for resources, modify soil chemistry or hydrology, alter fire regimes, or disrupt pollination and seed dispersal networks. Some invasive species exhibit allelopathic effects that directly inhibit native species through chemical interference. Management of invasive species requires understanding of invasion processes, including propagule pressure, invasion pathways, and factors that determine invasibility of different communities. Early detection and rapid response programs can prevent establishment of new invasive species, while integrated management approaches combining mechanical, chemical, and biological control methods may be necessary for managing established invasions. Prevention remains the most cost-effective approach, requiring regulation of trade pathways and education about invasion risks. Land use change continues to convert natural habitats to agricultural, urban, and industrial uses, directly eliminating plant communities and creating novel ecosystems with altered species compositions and ecological processes. Agricultural expansion remains a major driver of habitat loss worldwide, particularly in tropical regions where biodiversity is highest. Urban development creates novel environments with modified soils, altered hydrology, and unique microclimates that favor different species assemblages than natural habitats. The concept of novel ecosystems has emerged to describe communities that develop under altered environmental conditions or with modified species compositions that have no historical analogues. These systems may represent stable alternative states that persist even if historical disturbances are restored, requiring new approaches to conservation and management that focus on ecosystem function rather than historical species composition. Conservation strategies for plant communities in a changing world must address multiple threats simultaneously while recognizing the limitations of traditional approaches based on historical reference conditions. Protected area networks require expansion and improved connectivity to accommodate species migrations and maintain viable populations. Climate adaptation strategies may include assisted migration of threatened species, genetic rescue of small populations, and management interventions that facilitate ecosystem transitions to novel but functional states. Restoration ecology is evolving to address changing environmental conditions and the likelihood that historical reference conditions may no longer be achievable or appropriate. Forward-looking restoration approaches focus on establishing communities that can persist under projected future conditions rather than recreating historical states. This may involve using genetic material from warmer or drier regions, establishing communities with greater functional diversity, or creating habitat corridors that facilitate species movements. Ecosystem-based adaptation strategies recognize that healthy ecosystems provide crucial services for human communities while also supporting biodiversity conservation. These approaches integrate conservation goals with climate adaptation and mitigation objectives, recognizing that maintaining ecosystem integrity provides benefits for both human and natural communities. Examples include wetland restoration for flood control and carbon sequestration, forest management for watershed protection and carbon storage, and grassland conservation for soil health and wildlife habitat. The development of early warning systems for ecosystem change requires improved monitoring of plant communities and environmental conditions, combined with predictive models that can forecast likely changes under different scenarios. Remote sensing technologies enable monitoring of vegetation changes across large areas and over extended time periods, while permanent plot networks provide detailed information about community dynamics at local scales. Citizen science programs are becoming increasingly important for monitoring plant communities and documenting responses to environmental change. These programs can engage large numbers of volunteers in data collection activities while providing valuable information about species distributions, phenological changes, and invasion patterns. Training programs and standardized protocols ensure data quality while building public awareness of environmental change issues. International cooperation is essential for addressing global environmental challenges that affect plant communities, including climate change, trade in invasive species, and transboundary conservation issues. Global monitoring networks, such as the Global Biodiversity Information Facility and long-term ecological research networks, provide crucial data for understanding large-scale patterns and processes. International agreements and policies must address the root causes of environmental change while supporting local conservation efforts. The integration of traditional ecological knowledge with scientific approaches offers valuable insights for understanding and managing plant communities under changing conditions. Indigenous and local communities often possess detailed knowledge about plant ecology, traditional management practices, and ecosystem responses to environmental variation. Incorporating this knowledge into conservation and management planning can improve outcomes while respecting cultural values and traditional rights. Education and outreach efforts are crucial for building public support for plant conservation and ecosystem management. Understanding of plant ecology and the importance of plant communities for ecosystem services and human well-being remains limited among many segments of society. Effective communication about plant ecology must connect scientific findings with public values and concerns, while avoiding overwhelming audiences with complex information. The future of plant ecology depends on continued research, monitoring, and adaptive management approaches that can respond to rapidly changing conditions. Emerging technologies, including genomic tools, advanced remote sensing, and artificial intelligence, offer new opportunities for understanding and predicting ecosystem responses to environmental change. However, the ultimate success of conservation efforts will depend on addressing the underlying drivers of environmental change while building social and political support for effective action.
Chapter 1: The Mathematical Nature of Biological Systems. Quantitative biology represents a revolutionary approach to understanding life that bridges the traditionally descriptive nature of biological science with the precision and predictive power of mathematics and physics. This interdisciplinary field recognizes that biological phenomena, despite their apparent complexity and variability, follow quantifiable patterns and relationships that can be described, analyzed, and predicted using mathematical models and computational approaches. The emergence of quantitative biology reflects a fundamental shift in how we conceptualize living systems, moving from qualitative descriptions of biological processes to rigorous mathematical frameworks that reveal the underlying principles governing life at all scales, from molecular interactions within cells to population dynamics across ecosystems. The foundation of quantitative biology rests on the recognition that biological systems exhibit measurable properties that can be characterized using mathematical tools and statistical methods. Living organisms continuously process information, transform energy, and organize matter in ways that can be quantified and modeled. These processes often exhibit scaling relationships, where properties change in predictable ways across different scales of organization, revealing fundamental constraints and organizing principles that shape biological structure and function. Mathematical modeling in biology serves multiple purposes, from testing hypotheses about biological mechanisms to making predictions about system behavior under different conditions. Models can range from simple analytical expressions that capture essential features of biological processes to complex computational simulations that incorporate numerous interacting components and realistic environmental conditions. The power of mathematical modeling lies in its ability to distill complex biological phenomena into their essential elements, revealing the key factors that drive system behavior and enabling quantitative predictions that can be tested experimentally. The concept of biological scaling reveals how fundamental physical and chemical constraints shape the organization of living systems across different size scales. Allometric relationships describe how biological properties scale with organism size, often following power law relationships that reflect underlying geometric, physical, or physiological constraints. For example, metabolic rate typically scales with body mass raised to the power of 3/4 across many species, a relationship that emerges from the fractal-like organization of circulatory and respiratory systems that deliver nutrients and oxygen throughout the body. Surface area to volume ratios represent fundamental scaling constraints that influence many aspects of biological organization. As organisms increase in size, their volume increases faster than their surface area, creating challenges for processes that depend on surface exchange, such as gas exchange, nutrient uptake, and heat dissipation. These constraints drive the evolution of specialized structures, such as lungs, gills, and circulatory systems that increase effective surface area and enable efficient transport throughout large organisms. The application of thermodynamics to biological systems provides a quantitative framework for understanding energy flow and transformation in living organisms. The laws of thermodynamics impose fundamental constraints on biological processes, determining the efficiency of energy conversion, the direction of spontaneous processes, and the energy costs of maintaining biological organization. Biological systems appear to violate the second law of thermodynamics by creating and maintaining highly ordered structures, but this apparent contradiction is resolved by recognizing that organisms are open systems that exchange energy and matter with their environment. Information theory provides powerful tools for quantifying the information content and processing capabilities of biological systems. DNA sequences can be analyzed using information-theoretic measures to identify patterns, redundancy, and evolutionary signals. The information processing capabilities of neural networks, gene regulatory circuits, and other biological systems can be characterized using concepts such as mutual information, entropy, and channel capacity, revealing fundamental limits on biological computation and communication. Network theory has emerged as a crucial framework for understanding the organization and function of complex biological systems. Biological networks, including protein interaction networks, metabolic networks, gene regulatory networks, and ecological food webs, exhibit characteristic topological properties such as small world structure, scale-free degree distributions, and modular organization. These network properties have important implications for system robustness, efficiency, and evolutionary dynamics. The scale-free nature of many biological networks, where a few highly connected nodes, hubs, coexist with many poorly connected nodes, has important implications for system function and vulnerability. Hub nodes often play crucial roles in network function and may represent evolutionary bottlenecks or targets for therapeutic intervention. The removal of hub nodes can have disproportionate effects on network function, while the removal of randomly selected nodes typically has minimal impact. Stochastic processes play fundamental roles in biological systems, from the random nature of molecular collisions and chemical reactions to the unpredictable elements of evolutionary change and ecological dynamics. Understanding the sources and consequences of biological noise requires mathematical tools from probability theory and stochastic processes. Biological systems have evolved sophisticated mechanisms for dealing with noise, including error correction systems, redundancy, and robust network architectures that maintain function despite random fluctuations. The concept of emergence describes how complex biological phenomena arise from simpler underlying components and interactions. Emergent properties cannot be predicted solely from knowledge of individual components, but arise from the collective behavior of interacting systems. Mathematical models of emergence often reveal how simple local rules can generate complex global patterns, as seen in flocking behavior, cellular automata, and self-organizing systems. Optimization principles provide powerful frameworks for understanding biological design and function. Natural selection can be viewed as an optimization process that maximizes fitness subject to various constraints, leading to the evolution of efficient solutions to biological challenges. Mathematical optimization theory helps identify the trade-offs and constraints that shape biological systems and can predict optimal strategies for different environmental conditions. The application of control theory to biological systems reveals how organisms maintain homeostasis and respond to environmental perturbations. Biological control systems often exhibit sophisticated feedback mechanisms that maintain key variables within narrow ranges despite external disturbances. Understanding these control mechanisms requires mathematical tools from engineering and systems theory, including concepts such as stability, sensitivity, and robustness. Dynamical systems theory provides tools for understanding how biological systems change over time, including concepts such as equilibria, stability, bifurcations, and chaos. Many biological processes exhibit nonlinear dynamics that can lead to complex temporal behavior, including oscillations, switching between different states, and sensitive dependence on initial conditions. Understanding these dynamics is crucial for predicting system behavior and designing effective interventions. The integration of experimental data with mathematical models represents a central challenge in quantitative biology. Modern high-throughput experimental techniques generate enormous datasets that require sophisticated statistical and computational methods for analysis and interpretation. Machine learning approaches are increasingly important for extracting patterns from complex biological data and for developing predictive models that can guide experimental design and therapeutic development. Parameter estimation and model validation represent crucial steps in developing useful mathematical models of biological systems. Biological parameters often vary among individuals, populations, and environmental conditions, requiring statistical approaches that account for uncertainty and variability. Model validation involves comparing model predictions with experimental observations and assessing the robustness of models to parameter uncertainty and structural assumptions. The philosophy of quantitative biology emphasizes the importance of combining mathematical rigor with biological insight to develop models that are both mathematically sophisticated and biologically relevant. Successful quantitative biology requires close collaboration between mathematicians, physicists, computer scientists, and biologists, each bringing essential expertise to address complex biological questions. This interdisciplinary approach is revealing new principles of biological organization and function, while providing practical tools for addressing challenges in medicine, agriculture, and environmental management. Chapter 2: Population Dynamics and Evolutionary Mathematics. Population dynamics represents one of the most successful applications of mathematical modeling to biological systems, providing quantitative frameworks for understanding how populations grow, decline, and interact with their environment and other species. The mathematical description of population growth begins with simple exponential and logistic models, but extends to complex multi-species systems that incorporate spatial structure, age structure, genetic variation, and stochastic effects. These models have proven invaluable for wildlife management, conservation biology, epidemiology, and understanding evolutionary processes, demonstrating how mathematical approaches can provide both theoretical insights and practical applications for managing biological systems. The exponential growth model represents the simplest mathematical description of population dynamics, assuming that per capita birth and death rates remain constant over time. This model predicts unlimited population growth at a rate determined by the intrinsic rate of increase, which reflects the difference between birth and death rates. While exponential growth is rarely observed in natural populations for extended periods, it provides a useful baseline for understanding population potential and can accurately describe population growth when resources are abundant and populations are small. The mathematical formulation of exponential growth, dn dt equals Rn, where n represents population size, t represents time, and r represents the intrinsic rate of increase, leads to the solution nt equals ne(rt), demonstrating how populations can grow rapidly when conditions are favorable. The intrinsic rate of increase depends on species-specific life history characteristics, including age at first reproduction, reproductive rate, and survival probability, providing connections between individual-level traits and population-level dynamics. The logistic growth model incorporates environmental resistance through a carrying capacity term that limits population growth as density increases. This model predicts S-shaped growth curves that begin with exponential growth at low densities but slow as populations approach the carrying capacity of their environment. The logistic equation, dN dt equals Rn1nk, where k represents carrying capacity, provides a simple but powerful framework for understanding density-dependent population regulation. The carrying capacity concept represents the maximum population size that can be sustained indefinitely in a given environment, determined by the availability of limiting resources such as food, water, nesting sites, or other essential requirements. Carrying capacity is not a fixed property of environments, but can vary with environmental conditions, resource management, and changes in species interactions. Understanding factors that determine carrying capacity is crucial for conservation and management applications. Age-structured population models recognize that individuals of different ages contribute differently to population growth through variation in survival probability and reproductive output. These models track the number of individuals in different age classes and use age-specific birth and death rates to predict population dynamics. Leslie matrices provide a mathematical framework for analyzing age-structured populations, enabling calculation of population growth rates, stable age distributions, and population projections under different demographic scenarios. The concept of reproductive value quantifies the expected future reproductive contribution of individuals of different ages, providing insights into which age classes are most important for population growth. Reproductive value typically increases from birth to the age of first reproduction, then declines as individuals age and have fewer remaining reproductive opportunities. Understanding reproductive value helps guide conservation efforts by identifying which life stages are most critical for population persistence. Life table analysis provides detailed descriptions of age-specific mortality and reproductive patterns, enabling calculation of important demographic parameters such as net reproductive rate, generation time, and life expectancy. Life tables form the foundation for demographic models and provide essential data for understanding population dynamics and evolutionary processes. Comparative analysis of life tables across species reveals general patterns in life history evolution and the trade-offs that shape demographic strategies. Metapopulation models address the spatial structure of populations by considering networks of local populations connected through dispersal. These models recognize that many species exist as collections of local populations that can go extinct and be recolonized by dispersal from other populations. The balance between local extinction and recolonization determines regional persistence, with important implications for conservation in fragmented landscapes. The basic metapopulation model assumes that local populations have equal extinction and colonization rates, leading to equilibrium between occupied and unoccupied patches. More complex models incorporate variation in patch size, isolation, and quality, revealing how landscape structure influences metapopulation persistence. These models have important applications for reserve design and understanding how habitat fragmentation affects species persistence. Source-sink dynamics represent an important extension of metapopulation theory, recognizing that some populations consistently produce emigrants, sources, while others require immigration to persist, sinks. The persistence of sink populations depends on continued immigration from source populations, creating asymmetric dependencies that influence regional population dynamics. Understanding source-sink relationships is crucial for conservation planning and predicting responses to habitat loss or environmental change. Stochastic population models incorporate random variation in birth rates, death rates, and environmental conditions, recognizing that real populations are subject to unpredictable fluctuations that can have important consequences for population persistence. Demographic stochasticity arises from random variation in individual birth and death events and becomes increasingly important in small populations where chance events can have disproportionate effects on population size. Environmental stochasticity reflects random variation in environmental conditions that affect all individuals in a population, such as weather fluctuations, resource availability, or disturbance events. Environmental variation can be modeled as random fluctuations around mean conditions or as discrete environmental states with different population growth rates. Understanding environmental stochasticity is crucial for predicting extinction risk and designing robust conservation strategies. Population viability analysis combines demographic modeling with risk assessment to evaluate extinction probabilities and identify factors that threaten population persistence. These analyses typically incorporate multiple sources of uncertainty, including demographic stochasticity, environmental variation, and catastrophic events to estimate extinction probabilities over specific time horizons. Population viability analysis has become a standard tool for endangered species management and conservation planning. Evolutionary dynamics describe how gene frequencies change over time in response to natural selection, genetic drift, mutation, and gene flow. Population genetics provides mathematical frameworks for understanding these evolutionary processes, beginning with the Hardy-Weinberg equilibrium that describes gene frequency stability under simplified conditions. Deviations from Hardy-Weinberg predictions indicate evolutionary forces acting on populations. The fundamental theorem of natural selection states that the rate of increase in mean fitness of a population equals the additive genetic variance in fitness, providing a quantitative relationship between genetic variation and evolutionary change. This theorem demonstrates that evolution by natural selection tends to increase population fitness while reducing genetic variance, though the introduction of new mutations and changing environmental conditions can maintain genetic variation. Quantitative genetics extends evolutionary analysis to traits controlled by many genes, using statistical methods to partition phenotypic variation into genetic and environmental components. Heritability measures the proportion of phenotypic variation due to genetic differences and determines the response to selection. The breeder's equation, R equals H squared S, relates the response to selection, R, to the heritability H squared and selection differential, S, providing a predictive framework for evolutionary change. Game theory provides powerful tools for analyzing evolutionary strategies when fitness depends on the frequency of different strategies in the population. Evolutionary stable strategies represent equilibrium states where no alternative strategy can invade, providing insights into the evolution of behaviors, life history traits, and other frequency-dependent phenomena. The Prisoner's Dilemma and other classic games have been used to understand cooperation, conflict, and other evolutionary phenomena. Phylogenetic, comparative methods enable statistical analysis of evolutionary relationships and trait evolution across species. These methods account for the non-independence of species due to shared evolutionary history and enable tests of evolutionary hypotheses using comparative data. Phylogenetic methods have revealed general patterns in trait evolution and provided insights into the factors that drive evolutionary change. Molecular evolution describes changes in DNA and protein sequences over evolutionary time, providing insights into the mechanisms and patterns of evolutionary change at the molecular level. The neutral theory of molecular evolution proposes that most molecular changes are selectively neutral and evolve by genetic drift, leading to predictable patterns of sequence divergence that can be used for phylogenetic reconstruction and dating evolutionary events. Adaptive dynamics theory provides mathematical frameworks for understanding long-term evolutionary change when mutations have small effects on fitness. This approach can predict evolutionary trajectories, identify evolutionary endpoints, and reveal conditions for evolutionary branching that can lead to speciation. Adaptive dynamics has provided insights into the evolution of life history traits, cooperative behaviors, and other complex evolutionary phenomena. Coevolutionary dynamics describe evolutionary change in interacting species, where each species evolves in response to selection imposed by the other. Mathematical models of coevolution can lead to evolutionary arms races, cyclic dynamics, or stable coexistence, depending on the nature of the interactions and the genetic basis of relevant traits. Understanding coevolutionary dynamics is important for managing pest-crop interactions, disease dynamics, and conservation of mutualistic relationships. Chapter 3: Systems Biology and Network Analysis. Systems biology represents a paradigm shift in biological research, moving from reductionist approaches that focus on individual components to integrative approaches that examine how components interact to produce system-level properties and behaviors. This field employs mathematical modeling, computational analysis, and high-throughput experimental techniques to understand biological systems as networks of interacting components, revealing emergent properties that cannot be predicted from studying components in isolation. The mathematical foundations of systems biology draw from diverse fields, including graph theory, dynamical systems, control theory, and information theory, to provide quantitative frameworks for understanding complex biological networks. Biological networks represent the fundamental organizational structure underlying cellular and organismal function, with different types of networks governing distinct aspects of biological organization. Protein-protein interaction networks describe physical associations between proteins that enable the formation of functional complexes and signaling pathways. Metabolic networks map the biochemical reactions that transform nutrients into energy and cellular building blocks. Gene regulatory networks capture the control relationships between genes and their regulatory factors that coordinate cellular responses to environmental and developmental signals. Graph theory provides the mathematical foundation for analyzing biological networks, representing systems as collections of nodes connected by edges that represent interactions or relationships. Network topology describes the global structural properties of networks, including measures such as connectivity distribution, clustering coefficient, path length, and modularity. These topological properties have important implications for network function, robustness, and evolution. Scale-free networks exhibit degree distributions that follow power laws, where a few highly connected nodes coexist with many poorly connected nodes. Many biological networks display scale-free or approximately scale-free topology, including protein interaction networks, metabolic networks, and gene regulatory networks. The prevalence of scale-free organization in biological systems reflects evolutionary processes that favor the development of hub nodes that integrate multiple functions while maintaining network efficiency and evolvability. Small world networks combine high local clustering with short path lengths between distant nodes, enabling efficient information transmission while maintaining local specialization. This topology is common in neural networks, where local clusters of highly connected neurons coexist with long-range connections that integrate information across brain regions. Small world organization provides an optimal balance between global integration and local processing in many biological systems. Modular organization represents another fundamental property of biological networks, where nodes cluster into densely connected modules that are more sparsely connected to other modules. Network modules often correspond to functional units such as protein complexes, metabolic pathways, or regulatory circuits that carry out specific cellular functions. Modularity facilitates system evolvability by allowing independent modification of different functional units while maintaining overall system integrity. Centrality measures quantify the importance of individual nodes within network topology, providing insights into which components are most critical for network function. Degree centrality measures the number of connections per node, Betweenist centrality measures the extent to which nodes lie on paths between other nodes, and eigenvector centrality measures the influence of nodes based on their connections to other influential nodes. These measures help identify key regulatory nodes and potential targets for therapeutic intervention. Dynamic modeling of biological networks captures how network states change over time in response to internal dynamics and external perturbations. Ordinary differential equation models describe continuous changes in component concentrations or activities, while Boolean network models capture discrete switching behaviors in regulatory circuits. Stochastic models incorporate random fluctuations that can have important effects on network behavior, particularly in small systems where noise can drive transitions between different states. Gene regulatory networks control cellular responses to environmental conditions and developmental signals through complex circuits of transcription factors, signaling proteins, and regulatory RNAs. Mathematical models of gene regulatory networks reveal how simple regulatory motifs, such as feedback loops, feed-forward loops, and switches, can generate complex dynamic behaviors including oscillations, bistability, and adaptation. Understanding these regulatory circuits is crucial for predicting cellular responses and designing synthetic biological systems. Metabolic networks describe the biochemical reactions that convert nutrients into energy and biosynthetic precursors required for cellular growth and maintenance. Constraint-based modeling approaches, such as flux balance analysis, use stoichiometric constraints and optimization principles to predict metabolic flux distributions under different conditions. These approaches have proven valuable for understanding metabolic capabilities, identifying drug targets, and engineering microorganisms for biotechnology applications. Signaling networks transmit information about environmental conditions and internal cellular states through cascades of protein interactions and chemical modifications. Mathematical models of signaling networks reveal how cells can achieve specificity, sensitivity, and integration of multiple signals through network topology and dynamic properties. Signaling networks often exhibit ultra-sensitive responses, signal amplification, and adaptation mechanisms that enable robust cellular decision-making. Network motifs represent recurring patterns of connectivity that appear more frequently in biological networks than would be expected by chance. Common motifs include feedback loops that provide stability or oscillatory behavior, feed-forward loops that enable temporal filtering and logic operations, and regulatory cascades that enable signal amplification and fine-tuning. The prevalence of specific motifs in different types of biological networks provides insights into the selective pressures that shape network evolution. Robustness represents a fundamental property of biological networks that enables continued function despite perturbations such as component failures, environmental changes, or genetic mutations. Mathematical analysis reveals that network topology plays a crucial role in determining robustness, with redundant pathways, modularity, and distributed control contributing to system resilience. Understanding robustness is important for predicting system responses to perturbations and designing robust engineered systems. Network evolution describes how biological networks change over time through processes such as gene duplication, horizontal gene transfer, and regulatory evolution. Mathematical models reveal how network topology constrains evolutionary possibilities, while selection for network function drives the evolution of specific organizational principles. Comparative analysis of networks across species provides insights into universal principles of network organization and evolution. Control theory provides frameworks for understanding how biological systems maintain homeostasis and coordinate responses to environmental changes. Biological control systems often exhibit feedback regulation that maintains key variables within narrow ranges despite external disturbances. Mathematical analysis of control systems reveals trade-offs between different performance objectives, such as stability, responsiveness, and energy efficiency that shape the evolution of biological control mechanisms. Information processing in biological networks can be analyzed using concepts from information theory, including measures of information content, transmission, and integration. Neural networks process sensory information and generate appropriate behavioral responses through complex patterns of connectivity and dynamics. Gene regulatory networks process developmental and environmental information to generate appropriate patterns of gene expression. Understanding information processing capabilities helps reveal the computational principles underlying biological function. Synthetic biology applies systems biology principles to design and construct artificial biological systems with novel functions. This field requires quantitative understanding of biological components and their interactions to enable predictable engineering of biological systems. Mathematical modeling plays a crucial role in synthetic biology by enabling design of genetic circuits, prediction of system behavior, and optimization of engineered systems for specific applications. Network medicine applies network analysis to understand disease mechanisms and identify therapeutic targets. Disease networks reveal how genetic variants, protein interactions, and cellular pathways contribute to pathological states. Network-based approaches have identified disease modules, predicted drug targets, and revealed mechanisms of drug action and side effects. Understanding disease networks is crucial for developing more effective and targeted therapeutic strategies. Machine learning approaches are increasingly important for analyzing complex biological networks and extracting predictive insights from large-scale data. Deep learning methods can identify patterns in network structure and dynamics that are not apparent through traditional analysis methods. These approaches are particularly valuable for integrating diverse data types and generating predictive models of network behavior under different conditions. Chapter 4: Bioinformatics and Computational Methods. Bioinformatics represents the intersection of biology, computer science, and mathematics, developing computational methods to store, analyze, and interpret the vast amounts of biological data generated by modern experimental techniques. This field has become essential for biological research as high-throughput technologies produce datasets that are impossible to analyze without sophisticated computational approaches. The mathematical foundations of bioinformatics encompass algorithms for sequence analysis, structural prediction, phylogenetic reconstruction, and statistical analysis of large-scale biological data, providing tools that have revolutionized our understanding of molecular biology, evolution, and system-level biological processes. Sequence analysis represents one of the foundational areas of bioinformatics, developing algorithms to compare, align, and analyze DNA, RNA, and protein sequences. The basic problem of sequence alignment involves finding the optimal arrangement of sequences to maximize similarity while accounting for evolutionary processes such as mutations, insertions, and deletions. Dynamic programming algorithms provide mathematically rigorous solutions to alignment problems, guaranteeing optimal solutions while maintaining computational efficiency for practical applications. The Needleman-Wunsch algorithm implements global sequence alignment by creating a matrix of alignment scores and using dynamic programming to find the optimal alignment path. This algorithm considers all possible alignments and selects the one with the highest score according to a specified scoring system that rewards matches and penalizes mismatches and gaps. The mathematical elegance of this approach lies in its ability to decompose a complex optimization problem into simpler subproblems that can be solved systematically. Local sequence alignment addresses the problem of finding similar regions within sequences that may otherwise be quite different, reflecting the biological reality that functionally important domains may be conserved while other regions diverge rapidly. The Smith-Waterman algorithm extends dynamic programming principles to local alignment by allowing alignment scores to reset to zero, preventing negative scores from propagating and enabling identification of high-scoring local alignments. BLAST, Basic Local Alignment Search Tool, represents a heuristic approach to sequence searching that sacrifices guaranteed optimality for dramatic improvements in computational speed, enabling searches against large databases in reasonable time. The mathematical foundation of BLAST involves statistical analysis of alignment scores to determine the significance of matches, using extreme value statistics to calculate the probability that observed similarities could arise by chance. Multiple sequence alignment extends pairwise alignment to simultaneous alignment of many sequences, revealing evolutionary relationships and identifying conserved regions that may indicate functional importance. The computational complexity of optimal multiple alignment grows exponentially with the number of sequences, necessitating heuristic approaches such as progressive alignment that build multiple alignments by combining pairwise alignments in a systematic manner guided by phylogenetic relationships. Hidden Markov models provide probabilistic frameworks for modeling sequence patterns and predicting functional features such as genes, protein domains, and regulatory elements. These models represent sequences as paths through states that emit symbols with specific probabilities, enabling flexible modeling of biological sequence patterns while providing efficient algorithms for parameter estimation and sequence analysis. The Viterbi algorithm provides dynamic programming solutions for finding the most probable state path given an observed sequence. Phylogenetic analysis uses mathematical methods to reconstruct evolutionary relationships among species or genes based on sequence similarity patterns. Distance-based methods such as neighbor joining use pairwise evolutionary distances to construct trees that minimize total branch length. Character-based methods, such as maximum parsimony and maximum likelihood, evaluate specific evolutionary scenarios and select trees that best explain observed sequence patterns according to explicit evolutionary models. Maximum likelihood approaches to phylogenetic reconstruction use statistical models of sequence evolution to calculate the probability of observing specific sequence patterns given particular tree topologies and evolutionary parameters. These methods provide statistically rigorous frameworks for tree reconstruction, while enabling testing of evolutionary hypotheses and estimation of evolutionary parameters such as substitution rates and divergence times. Bayesian phylogenetic methods extend maximum likelihood approaches by incorporating prior distributions over tree topologies and evolutionary parameters, enabling estimation of uncertainty in phylogenetic reconstructions. Markov chain Monte Carlo algorithms provide computational methods for sampling from complex posterior distributions, enabling exploration of tree space and quantification of phylogenetic uncertainty. Genome assembly represents a fundamental computational challenge in bioinformatics, involving the reconstruction of complete genome sequences from short DNA fragments produced by sequencing technologies. The mathematical complexity of genome assembly arises from the presence of repetitive sequences that create ambiguities in fragment placement, requiring sophisticated algorithms that can resolve these ambiguities using overlapping information and paired-end constraints. De Bruijn graphs provide mathematical frameworks for genome assembly by representing overlaps between sequence fragments as paths through graphs, where nodes represent short sequences and edges represent overlaps. These graphs enable efficient identification of paths that correspond to genomic sequences, while providing algorithms for resolving complex patterns of repeats and structural variations. Gene prediction algorithms identify protein-coding genes and other functional elements within genome sequences using computational models that recognize characteristic sequence patterns. Hidden Markov models and other machine learning approaches can be trained on known gene structures to recognize patterns such as start codons, splice sites, and stop codons that define gene boundaries. Comparative approaches use evolutionary conservation across species to improve gene prediction accuracy. Structural bioinformatics applies computational methods to predict and analyze three-dimensional protein structures from amino acid sequences. Homology modeling uses known protein structures as templates to predict structures of related proteins, relying on the principle that similar sequences tend to adopt similar structures. The mathematical challenges involve optimizing atomic coordinates to minimize energy functions while satisfying geometric constraints imposed by chemical bonds and physical interactions. Ab initio protein structure prediction attempts to predict protein structures directly from amino acid sequences using physical principles and statistical potentials derived from known structures. These approaches face enormous computational challenges due to the vast number of possible conformations that proteins can adopt, requiring sophisticated sampling algorithms and energy functions that can distinguish native-like structures from incorrect folds. Molecular dynamics simulations use classical mechanics to model the motion of atoms in biological molecules over time, providing insights into protein flexibility, binding mechanisms, and other dynamic processes that are crucial for biological function. These simulations require numerical integration of Newton's equations of motion for systems containing thousands or millions of atoms, demanding sophisticated algorithms and high-performance computing resources. Machine learning approaches have become increasingly important in bioinformatics, providing powerful tools for pattern recognition, classification, and prediction tasks. Support vector machines, neural networks, and other machine learning algorithms can identify complex patterns in biological data that are difficult to capture with traditional statistical methods. Deep learning approaches have shown particular promise for problems such as protein structure prediction and gene expression analysis. Statistical analysis of high-throughput biological data requires specialized methods that can handle the challenges of large numbers of variables, small sample sizes, and multiple testing problems. Microarray and RNA sequencing data analysis involves normalization procedures to remove technical artifacts, statistical tests to identify differentially expressed genes, and multiple testing corrections to control false discovery rates. Network analysis methods enable investigation of complex relationships in biological data, revealing modules of co-regulated genes, protein interaction networks, and regulatory circuits. Graph-based clustering algorithms can identify functional modules within networks, while centrality measures can identify key regulatory nodes that may represent important therapeutic targets. Integration of diverse data types represents a major challenge in computational biology, requiring methods that can combine information from genomics, transcriptomics, proteomics, and other high-throughput technologies. Multidimensional scaling, principal component analysis, and other dimensionality reduction techniques enable visualization and analysis of complex multidimensional datasets while preserving important relationships among variables. Database design and management represent crucial infrastructure components of bioinformatics, requiring efficient storage and retrieval systems for large biological datasets. Relational databases, NoSQL systems, and specialized biological database formats must balance storage efficiency, query performance, and data integrity while supporting the complex relationships inherent in biological data. Cloud computing and distributed systems have become essential for handling the computational demands of modern bioinformatics, enabling analysis of large datasets that exceed the capabilities of individual computers. Parallel algorithms and workflow management systems enable efficient distribution of computational tasks across multiple processors while maintaining data integrity and result reproducibility. Quality control and validation represent critical aspects of bioinformatics analysis, requiring statistical methods to assess data quality, identify potential artifacts, and validate computational predictions. Cross-validation, bootstrapping, and other resampling methods enable assessment of prediction accuracy and uncertainty, while benchmark datasets provide standardized evaluation criteria for comparing different computational methods. Chapter 5: Mathematical Modeling in Medicine and Drug Development. Mathematical modeling has emerged as an indispensable tool in modern medicine and pharmaceutical development, providing quantitative frameworks for understanding disease mechanisms, predicting treatment outcomes, and optimizing therapeutic strategies. The application of mathematical principles to biomedical problems encompasses diverse areas, including pharmacokinetics and pharmacodynamics, epidemiology, clinical trial design, and personalized medicine. These quantitative approaches enable researchers and clinicians to move beyond descriptive observations to predictive understanding, facilitating the development of more effective treatments while reducing the time and cost associated with medical research and drug development. Pharmacokinetic modeling describes the time course of drug concentrations in the body, encompassing the processes of absorption, distribution, metabolism, and elimination that determine how drugs reach their targets and how long they remain active. The mathematical foundation of pharmacokinetics relies on differential equations that describe drug movement between different body compartments, with rate constants that characterize the efficiency of various physiological processes. These models enable prediction of drug concentrations at any time following administration and optimization of dosing regimens to achieve desired therapeutic effects while minimizing adverse reactions. The simplest pharmacokinetic model treats the body as a single well-mixed compartment where drug concentration decreases exponentially following administration due to first-order elimination processes. This one-compartment model, described by the equation dC dt equals Kc, where C represents drug concentration and K represents the elimination rate constant, provides useful approximations for many drugs and enables calculation of important parameters such as half-life and clearance that guide clinical dosing decisions. Multi-compartment models recognize that drugs distribute to different tissues at different rates, creating complex patterns of concentration change over time. Two-compartment models distinguish between central compartments representing blood and highly perfused tissues that equilibrate rapidly with administered drugs, and peripheral compartments representing less well-perfused tissues that equilibrate more slowly. These models can capture the initial rapid decline in drug concentration, followed by slower terminal elimination phases, commonly observed with many medications. Physiologically based pharmacokinetic models represent the most sophisticated approach to drug disposition modeling, incorporating detailed anatomical and physiological information about organ sizes, blood flows, and tissue binding properties. These models enable prediction of drug concentrations in specific organs and tissues, facilitating understanding of target site exposure and potential toxicity. The mathematical complexity of these models requires computational methods to solve systems of differential equations describing drug movement throughout the body. Pharmacodynamic modeling describes the relationship between drug concentrations and pharmacological effects, bridging the gap between pharmacokinetics and clinical outcomes. The Hill equation, E equals Emax times C caret 10, EC50 caret 10 plus C caret 10, where E represents effect, C represents concentration, Emax represents maximum effect, EC50 represents the concentration producing half maximal effect, and N represents the Hill coefficient, provides a flexible framework for describing concentration effect relationships with different shapes and sensitivities. The integration of pharmacokinetic and pharmacodynamic models enables prediction of drug effects over time following administration, accounting for both the time course of drug exposure and the relationship between exposure and response. These PK/PD models are essential for optimizing dosing regimens, particularly for drugs with narrow therapeutic windows where small changes in dose can dramatically affect efficacy and safety. Population pharmacokinetic and pharmacodynamic modeling recognizes that drug responses vary among individuals due to factors such as age, weight, genetic polymorphisms, and disease states. These models use statistical methods to characterize typical parameter values and inter-individual variability, enabling identification of factors that influence drug response and development of individualized dosing recommendations. Mixed effects modeling provides mathematical frameworks for analyzing data from multiple individuals while accounting for both fixed effects that apply to all individuals and random effects that vary among individuals. Epidemiological modeling applies mathematical approaches to understand the spread of diseases through populations and evaluate intervention strategies. The basic reproduction number R0 represents a fundamental parameter that determines whether an epidemic will spread, calculated as the average number of secondary infections caused by a single infected individual in a completely susceptible population. When R0 exceeds 1, epidemics can occur, while values below 1 indicate that diseases will die out. Compartmental models in epidemiology divide populations into categories such as susceptible, infected, and recovered individuals, with differential equations describing transitions between compartments. The SIR model represents the simplest framework, assuming that susceptible individuals become infected at a rate proportional to their contact with infected individuals, and infected individuals recover at a constant rate. More complex models incorporate additional compartments, such as exposed individuals in SEIR models or vaccinated individuals in SVIRV models. Spatial epidemiological models extend basic compartmental approaches to account for geographic spread of diseases through transportation networks, population movements, and spatial clustering. These models are essential for understanding pandemic spread and evaluating geographically targeted intervention strategies such as travel restrictions or regional vaccination campaigns. Network models represent populations as nodes connected by links representing potential transmission pathways, enabling analysis of how network structure influences disease spread. Stochastic epidemiological models incorporate random variation and transmission events, particularly important for understanding disease dynamics in small populations where random effects can significantly influence outcomes. These models can predict extinction probabilities for disease outbreaks and identify conditions under which diseases may persist in populations. Monte Carlo simulation methods enable analysis of complex stochastic epidemic models that cannot be solved analytically. Clinical trial design relies heavily on statistical methods to ensure that studies can detect meaningful treatment effects while controlling the probability of false positive and false negative results. Sample size calculations use statistical power analysis to determine the number of participants needed to detect clinically relevant differences between treatment groups with specified levels of confidence. These calculations must account for expected effect sizes, variability in outcomes, and acceptable levels of statistical significance and power. Adaptive clinical trial designs use accumulating data to modify study conduct while maintaining statistical validity, potentially reducing sample sizes and accelerating drug development. Bayesian methods provide frameworks for incorporating prior information and updating beliefs about treatment effects as data accumulate. Sequential designs enable early stopping for efficacy or futility based on interim analyses, while adaptive randomization can increase the probability that participants receive more effective treatments. Survival analysis methods are crucial for analyzing time-to-event data common in medical research, such as time to disease progression, treatment failure, or death. The Kaplan-Meier method provides non-parametric estimates of survival functions, while Cox proportional hazards models enable analysis of factors that influence survival times. These methods must account for censoring that occurs when observation periods end before events occur, requiring specialized statistical techniques. Personalized medicine represents an emerging application of mathematical modeling that aims to tailor treatments to individual patients based on their specific characteristics. Predictive models use patient data, including genetic information, biomarkers, and clinical characteristics, to estimate the probability of treatment success or adverse events. Machine learning approaches can identify complex patterns in patient data that predict treatment responses, enabling more precise therapeutic decision-making. Pharmacogenomic modeling describes how genetic variations influence drug responses, enabling prediction of optimal doses for individual patients based on their genetic profiles. These models must account for the complex relationships between genotype and phenotype, often involving multiple genes and their interactions. Population genetics principles help predict the distribution of relevant genetic variants in different populations and guide the development of genotype-based dosing guidelines. Systems pharmacology approaches integrate mathematical models of drug action with models of disease mechanisms to understand how drugs affect complex biological networks. These models can predict drug effects on multiple targets and pathways, enabling identification of biomarkers for drug response and potential side effects. Network-based approaches reveal how drugs influence interconnected biological systems and may identify combination therapies that target multiple disease mechanisms. Optimal control theory provides mathematical frameworks for determining treatment strategies that optimize patient outcomes while minimizing costs or side effects. These approaches treat medical interventions as control inputs that can be adjusted over time to achieve desired therapeutic goals. Applications include optimal dosing strategies for chemotherapy, timing of interventions in infectious diseases, and resource allocation in healthcare systems. Cost-effectiveness analysis uses mathematical modeling to evaluate the economic value of medical interventions, comparing costs and health outcomes across different treatment strategies. Markov models are commonly used to simulate disease progression and treatment effects over extended time periods, enabling calculation of quality-adjusted life years and cost-effectiveness ratios. These analyses inform healthcare policy decisions and reimbursement determinations. Regulatory science applies mathematical modeling to support drug approval decisions by quantifying benefits and risks of new treatments. Model-based approaches can integrate data from multiple studies, predict drug performance in different populations, and support labeling decisions. Regulatory agencies increasingly accept model-based analyses as evidence for drug approval, particularly when traditional clinical trials are not feasible. Digital therapeutics represent an emerging field that applies software and digital technologies as medical interventions, often incorporating mathematical models to personalize treatment delivery. These applications use behavioral models to optimize intervention timing and intensity, machine learning to adapt to individual patient responses, and control theory to maintain therapeutic engagement. The quantitative nature of digital therapeutics enables precise measurement of intervention effects and continuous optimization of treatment protocols.